Hello, my dear students, and welcome back to Victory Batch. And I am your Diksha ma'am. So, today we are going to start with the chapter Locomotion and Movement. And I hope you must have completed the entire chapters which I have covered here. So, it's time to start with the chapter that helps, or your body physiology parts that helps you to move. So, let's get started. So, first of all, let's talk about what is the meaning of this name of the chapter. Locomotion and movement. What is a locomotion and what is a movement? There is a fine line, a very small difference between the two, but it creates a large difference, right, in the terms of their physiology. So if I talk about, I am moving my arm, but I'm standing at this position only and I'm not changing my position. This is what movement is, right? Or for example, I'm also moving my leg. You just can't see it. Otherwise, I have to pick up my leg and that's I cannot do, right? So I am moving my leg, but I'm standing on the same position. This is what is movement is. In the movement, your muscles are working. Yes, your muscles are working, but you're just standing at one position. But with the same leg, I am also changing my position and I am going from one place to another like this. This is what locomotion is. This is the difference between locomotion and movement. The same organs are working for both, but the only difference is in locomotion, you can move, but in movement, you're just standing at one position. So we say that all locomotions are movement because whenever I'm doing the locomotion, I'm also doing the movement. Right, but all movement cannot be locomotion because in locomotion you are just standing at one particular position, right? So we say that what is a locomotion? It is the change in, position of entire body from one place to another, from one place to another, right? Whereas if I talk about movement, movement is just you're moving your body part, right? So it's just the movement or the displacement of a body part. But no change in position. Fine. So this is how we say that what's that golden line? These lines are also important. These can be, sometimes, examiners, these NTA who make this question paper of NEET. Nowadays, they are putting out a question of a kind where they will directly give the lines of NCERT. So you need to know what is the meaning of the line, whether it is right or wrong. Okay. So there's a line of NCERT, all locomotions are movement, but all movements are not locomotion. All right. Now, as I say that the same body organ can cause locomotion as well as movement. So one such example is your skeletal muscle. Like in human beings, skeletal muscles are also helping in the movement and it is also helping in the locomotion. What about the other organisms? In other organisms, like we have paramecium. So you must have heard of paramecium. I don't think so there will be any student who don't know about paramecium because you cover this in the botany section, right? And then we have this organism that is the hydra. Now, just like human beings, okay, I'm also mentioning here human beings. In human, the muscles, they are causing locomotion and also they are causing movement. Fine. All right, for example, the visceral muscle or smooth muscles in your intestine, they're causing movement of the food, whereas skeletal muscles are helping you to locomotion. Also, skeletal muscles are also helping you to move. Fine. Okay, now, how about the paramecium? In paramecium, we have the structures, the hair-like structures known as cilia, and in hydra, we have these structures known as tentacles. Okay. Now, like in us, the muscles are helping in the movement, in paramecium, the cilia, and in the hydra, tentacles. So, as you can see, this is the body of your, what do you say, paramecium. In paramecium, in this oropharynx. Oropharynx is a portion where your food enters. Oropharynx have cilia. So this cilia in the oropharynx, which later form the cytopharynx, okay, when the food gets involved with that. So this one is leading to the movement of food. So cilia in the oropharynx, it helps in movement of food. So this is what it is doing, the movement. Okay, so as I've said, the cilia and paramecium is helping in both locomotion and movement. So this is what about the movement. The movement of food is also taken place, or it takes place with the help of cilia present in the oropharynx. Now, also it has cilia across the body, and this cilia will help in the locomotion. That means it will help the paramecium to displace from one position to another. Okay. Now, what about tentacles? So these structures are tentacles in the hydra. These tentacles, they also helps in the movement. How about movement? In movement, the tentacles, for example, the hydra is just standing on one surface attached to the substratum. Now it has to capture the food from here. So this is what movement is, but it is not changing its position. So with the help of tentacles, the hydra can catch the prey or food, right? So what about movement? It helps in the catching of food. It catches the food. If I talk about the locomotion by the tentacles, yes, it helps it to move from one place to another. It also helps the hydra to swim. Yes, so because hydra is present in the water, so it can help the hydra to swim as well. Fine, guys. So that's about how one structure can lead to both locomotion and movement. Now let's talk about some lines of NCERT here, because I feel that NCERT is very important. So those who do not have a habit of reading NCERT's first or second paragraph, Diksha ma'am is here. She will not leave you like that. There is a habit of students. They will always skip the first paragraph of NCERT. No, that's the paragraph that makes the base of the chapter. Always read that, right? So let's get started. Movement is one of the significant feature of living beings. We know this is a significant feature especially of the animals. Locomotion is a feature of animals, not the plants. Plants cannot move from one place to another, but animals can. Animals and plant exhibit a wide range of movement. Streaming of protoplasm in the unicellular organism like amoeba is the simplest form of movement. So plants cannot undergo locomotion, but yes, they can undergo movement. That you must have studied in the plants. Like there is a plant like Mumisa pudica, if I'm not wrong, that chui mui in Hindi we say, touch me not plant. That if you touch it, it will collapse and take away its the leaves gets towards inside. It will not bloom, right? So this is the movement which is shown by the plants. Okay, then movement of cilia. Okay, then about amoeba. So amoeba is also a unicellular organism, and you all know how does amoeba move. Amoeba moves by formation of these structures known as pseudopodia. These are what? Pseudo means false feet. So this is how this unicellular organism amoeba moves. Okay, now next line. Movement of cilia, flagella and tentacles are shown by many organisms, like cilia by the, you all know, cilia by paramecium, flagella, euglena, and the tentacles, hydra. Human beings can move limbs, jaws, eyelids, tongue, etc. Right now, I am doing all that. Right, I'm also moving my lips, I'm also moving my jaws, I'm also moving my eyelids, and I am also moving my tongue. This is how I speak, right? Some of the movements result in a change of place or locomotion. Such voluntary movements are called locomotion. So any movement that can cause a change in a position, what you call it as locomotion. Walking, running, climbing, flying, swimming are all some forms of locomotory movement. Locomotory structure need not to be different from those affecting others by the movement. As I have already told you, locomotion and movement is caused by the same organ. This is what this line says in a bit of an English, greater English form. For example, in paramecium, cilia helps in the movement of food through the cytopharynx. As I've told you, this oropharynx, when it gets food, you can also call it as oropharynx. Okay? Oh, sorry, cytopharynx. Next. And in locomotion as well. Hydra can use its tentacle for capturing its prey and also use for locomotion. We use limbs for change in the body posture and locomotion as well. The above observations suggest that movements and locomotion can be studied separately. The two may be linked by stating that all locomotions are movement, but all movements are not locomotion. Yes, you can study them differently because the locomotion can be caused by some other factors. Physics can be different, but biology or this muscle contraction will be same. Okay. Methods of locomotion performed by animals vary with their habitats and the demand of situation. Now, as you have seen, amoeba walks by the pseudopodia. Okay. And as we have discussed, paramecium, it walks by cilia. Hydra walks by, guys, tell me. Hydra walks by tentacles. Okay. And human, they walk by with the help of muscles. Now, why all these organisms, they are having different types of these locomotory structures? The reason is, first of all, as you can see here, the reason is, first of all, where they are present. Like the hydra, it's usually found in the water. So that's why they have certain kind of structure. Or we can say why fishes have fins, because they live in water. Do any terrestrial organisms have fins? Do we have fins? No, because we are land animals. First is habitat, where they present. On that, it depends that what kind of locomotory structure an organism can have. Okay, and yes, due to evolution, also due to habitat, the complexity of organism changes, and that can also change the organization of the body. Okay. Second is demand of the situation. What's the demand? For example, if I say fish has to swim, so it will have fins, and we have to walk. So we are terrestrial, we will have limbs, okay? Or just like frog has to walk, or reptiles have to walk. All these are terrestrial. So that's a situation where we live and what kind of a situation is we want to walk, so we have limbs. However, locomotion is generally for, why do we have locomotion? We have for search of food, shelter, mate, suitable breeding grounds, favorable climatic condition, or to escape from enemies or predators. Why do, with evolution, organisms have specialized limbs? For example, cheetah have limbs for running. Whale is a mammal, but it lives in water, so it has its fin-like structures, okay, like flippers. And bat is also mammal, it flies. We are human being, our limbs are adapted to grasp thing. We'll also study this in evolution. Grasp means to hold things, like I'm holding this pen, because this is how we have done evolution. This is how we have changed according to our situations, right? Now, why we have done this? To search for food. Because if we have limbs, if we can walk, only then we can go and search for food. Okay, but in human nowadays, the search for food is quite have a different statement. Search for job. If you'll go to work, only then you can earn your bread. This is what the search for food is, right? Shelter, to have a house, big house. If you'll go for search for a job, you will have the shelter, right? Mate, to find the good mate. For example, if the boy is not earning good, will any father give their daughter to that guy? No one will give. So so for better search for mate, also there are limbs given, so go use your limbs, do something and earn money. Suitable breeding grounds, favorable climatic conditions. For example, there is raining outside, you just can't, if you're not a plant, you just can't stand there still like that. You will use your limbs and go to some shade areas where there is a shade and house or something, right? Or to escape from enemies or predator. If, for example, if there is a cheetah roaming around outside, will you go outside? Or if you're already standing outside, you will run towards and go inside the house. So this is how you can also run away from the enemies, right? This is about all human beings, because that's relatable enough. So this is why every organism have different types of organs. Okay, then let's talk about types of movement. So movement can be of two types. It can be non-muscular, guys. That means there is no use of muscles. Or it can be muscular. That means muscles plays a vital role. Okay. So, first of all, in the non-muscular one, we have ciliary movement. And we have the pseudopodial movement. Pseudopodial, also known as amoeboid movement. Now here I'm just talking about human beings, nothing else, just human beings. Okay, so where do you have your ciliary movement? Do we have cilia? Yes, in the structural organization in animals, I have told you that we do have cilia, and that cilia is present in the respiratory tract and fallopian tube. So that cilia is leading to the movement of substances. In the respiratory tract, there is a movement of mucus and dust particles. In the fallopian tube, there is a movement of ova. So yes, we do perform ciliary movement where first in the fallopian tube. What are we moving in the fallopian tube? Ova or egg or gamete. And in the respiratory tract, what are we moving? We are moving, yes, the mucus and dust particles in a specific direction. So the position of cilia is in a such a way that it will push out or pull out the things in a specific direction. Okay. Then pseudopodial movement or amoeboid movement, yes, it is also seen in our body in macrophages and WBCs. Macrophages and WBCs. Guys, what are WBCs? White blood cell. So white blood cell or WBCs, they show amoeboid or pseudopodial movement. Talking about the muscular movement, we have three types of muscle. One is a skeletal, another is a visceral muscle, and third is a cardiac muscle. All these muscle leads to the movement. All are responsible for movement, but maximum movement is done by the skeletal muscle, and this is also responsible for locomotion. If I talk about which muscle is responsible for locomotion, then you will say skeletal. Which muscle is responsible for movement? You will say these all three. Okay, that's about what type of movements are there. So this is the movement that we need to discuss in the more detail. But before that, let's read this paragraph also. We don't have to leave any paragraph. Some specialized cells in a body like macrophage and leukocyte in blood exhibit amoeboid movement. So as I've told you, what cells exhibit amoeboid movement? Macrophages and leukocytes. It is affected by pseudopodia formed by the streaming of protoplasm. So now how does amoeba move? How does amoeba move? So amoeba have cytoplasm in it. Okay, so when it will move its cytoplasm in a certain direction. For example, if it will have, it will move its cytoplasm in this area, there will be the formation of a false feet known as pseudopodia. So yes, movement of cytoplasm is very important. Also, the cytoskeleton like microfilaments are involved in amoeboid movement. Now, why this structure has become a feet-like? Because here, a lot of microfilaments, they are giving or providing shape here. They are providing shape here, right? So we say that first, okay, we say that first, cytoplasmic movement, okay, cytoplasmic movement, also known as cytoplasmic streaming, and the microfilaments here, they are playing a vital role in the formation of this false feet, and this is how the amoeba moves. First it will form the feet here. Now it has to move in this direction, then it can also form a feet here in this direction also. So this is how your WBCs also walk, just like amoeba. Okay. Second, ciliary movement occur in most of our internal tubular organs which are lined by ciliated epithelium. So as I've told you, which epithelium is responsible? Ciliated, because that have cilia. The coordinated movement of cilia in the trachea help us in removing dust particles and some of the foreign substances inhaled along with the atmospheric air. So what's the purpose of respiratory tract having cilia? That any dust particle, it gets intact, or it it gets inhaled along with the air. You have to remove it out, and with the for removal of that dust particles, we need to have the cilia over there. So people who smokes a lot, their cilia, they got damaged, and that's why a lot of dust particles can go inside their lungs also. Okay, so never ever smoke. This is really bad. Passage of ova through the female reproductive tract is also facilitated by ciliary movement. So ova movement in the fallopian tube. Movement of our limbs, jaws, tongue, etc. required muscular movement. The contractile property of muscles are effectively used for locomotion and other movements by human being and majority of multicellular organism. Locomotion require a perfect coordinated activity of muscular, skeletal and neural system. In this chapter, you will learn about the types of muscle, their structure, mechanism of their contraction and important aspects of skeletal system. So this is what we have to study in the entire chapter. So let's get started with the other topics, guys. Okay. So I hope each line of NCERT is clear to you. Yes. Okay. So I hope everything is clear to you guys now. So we are going to start with the muscle part. as we have discussed also in the chapter structural organization in animals, but I will also discuss the same here also, because I don't want you to leave any topics behind. Okay, so those who have already attended and learned it, you can have a revision part with me in this muscle. Otherwise, you can also skip it if you want, right? But I will always suggest that revise it along with me. So let's get started with the muscles. So if I talk about muscles, guys, they are mesodermal in origin. What is the origin? Mesodermal. So they are mesodermal in origin. Mesoderm in origin. Okay. If I talk about your body weight, for example, if I say someone is 60 kgs, so 40 to 45% of that weight is the muscular weight. Okay, so 40 to 45%, 40 to 50% of body weight is your muscle. All right, guys. Next, if I talk about the muscle, what are the various properties your muscles have? Your muscles can contract and relax. That's a property of contractility or contractibility. Second, just like that of neuron, they are also excitable. So excitability. It can also extend. So it has a property of extensibility. Right? It can extend, right? And the last property, just like a rubber band, it has a property of elasticity. That's why you are flexible. Okay, so flexibility was because of the joints, muscles, and cartilages, and muscles are very important for movement. Okay, so basically the movement is not just done by the muscles. Efforts are made by muscles, but it's the joint action of your neural system, muscles, joints, and your skeletal system. Right? Okay, so these are the various properties of the muscle. So we have so far done three types of muscle. One is a skeletal, another is a smooth, also known as visceral, and third is a cardiac muscle. Let's talk about all these muscles in very much detail. Okay, so we'll start with the skeletal muscle. Skeletal muscles are voluntary muscles. Why they are known as voluntary muscles? Because they are under your control, under your will. Whenever you want to, you can just move it according to your will, right? Second, they are also known as striped muscle or striated muscle because they have alternative light or dark bands. So we'll get started with this one. First of all, why it is known as a skeletal muscle? Because it is attached to bones. It is attached to bones. Apart from that, it is also present in the tongue, upper 1/3 of esophagus. They are your skeletal muscle. They are also present in the sphincters, like we have done in the urethral sphincter. We have one of a smooth muscle, another of a skeletal muscle. Okay, and also it's present in your body wall. In your body wall. Okay. Second, we say it's voluntary. It is under our control. It is under our control, and this is how you call it as it is voluntary. So whenever I want, I can move my arms. This is what voluntary is. Third, we have it has striations. It has striations, and these striations are usually due to alternative light and dark bands. Alternative light and dark bands. These alternative light and dark bands, these are formed of the contractile proteins. What are they made up of? Made of contractile proteins. Contractile proteins, myofilaments. Okay. All right. So let's talk about the structure of the muscle now. If I talk about the structure of the skeletal muscle, how does it looks like? Imagine, guys. Okay, let me just draw this one. Imagine this is a transverse section. Imagine this is a transverse section of your muscle. Okay, this is one muscle. So what is present inside the muscle? Let's see. So muscle is basically made up of small muscle bundles. Small muscle bundles, also known as muscle fascicles. also known as muscle fascicle. So this entire red is your muscle. This is a transverse section of your muscle. Which muscle? Transverse section of your skeletal muscle. Okay, this is your skeletal muscle. So here you can see this skeletal muscle. Inside this are present these structures known as muscle bundles or muscle fascicles. Fascicles. Okay, now these muscle bundles and muscle fascicle, they can move here and there. So there is a connective tissue present between them. So there is a connective tissue that is present between them like this. So you call this connective tissue as fascia. What do you call it as? Fascia. So this is fascia. So fascia is a connective tissue. It's a connective tissue that holds all the muscle bundles together. That holds all muscle bundle together. Can you guess what type of connective tissue this can be? Yes? Just have a rough guess. Okay, I'll give you a clue. This is binding all the organs together. So which connective tissue binds all the organs together? That is areolar connective tissue. Okay, all right. So next we have what is present inside the muscle bundles. So inside the muscle bundles, what are present? Inside the muscle bundles are present small muscle cells. Okay, small these muscle cells or muscle fibers, muscle cells or fibers. As you can see, I have made the transverse section. So anything that appears circular in the transverse section, that things are generally cylindrical. So we say that muscle cells are also cylindrical. Skeletal muscle is also cylindrical. For example, if this is a muscle, it's cylindrical. If I take a transverse section, then here you can see it's circular. So that's why it appears circular, because these muscles are cylindrical in shape. So as you can see, these are muscle fibers of muscle cell. Inside them are present small myofibrils. What are these? Myofibrils. Okay, these are what? Myofibrils. Now let's draw the structure of one skeletal muscle cell or skeletal muscle fiber. Okay, all right guys. So as I've told you, this is cylindrical in shape. This is cylindrical in shape. This is how it is. It is cylindrical in shape. Like that. Okay. So the shape is, I'm writing side by side things, so you have to learn with me. Okay. What's the shape of the skeletal muscle cell? It's cylindrical. This is the most common question that can be asked in the exam. Second thing, it has a specialized plasma membrane. It has a specialized plasma membrane. You call it as sarcolemma. What do you call it as? Sarcolemma. So this is sarcolemma, guys. What is it? It's sarcolemma. Okay. So it has a specialized plasma membrane. What's the name of the plasma membrane? Sarcolemma. So yes, it also has sarcolemma. So let me just label this for you. This is sarcolemma. Next. The cytoplasm of skeletal muscle is now, it will not just be known as a cytoplasm. Now you have a special name for this, and that is sarcoplasm. So yes, it do have sarcoplasm. Okay, sarcoplasm is the cytoplasm. All right, so second point, sarcoplasm present. Okay, next, this muscle have nuclei, or this muscle cell have nuclei because it's a cell. But how many? Many. Yes, how many? Many. It has a number of nucleus, and all are present at the peripheral position. Peripheral means outside, like this, on the outer edges, not on the center. Usually a cell has nucleus in the center, but this one here, there are nuclei present, number of nuclei, and this kind of situation is known as syncytium. What is syncytium condition? Whenever I'm using the word syncytium, that means multinucleated condition, where one cell has been formed by the fusion of number of cells. For example, if you have 5 cells, if I fuse all the cells together, one cell will be formed. Then how many nucleus will be there in that cell? 5 nucleus. Okay, so we say that syncytium condition is formed here because it has a number of nuclei, many nuclei, and nuclei at periphery. So nuclei at peripheral position or periphery. Apart from that, guys, you can see here the plasma membrane or the sarcolemma has been invaginated and it is forming the structure known as T-tubules. What's the function of these T-tubules? Inside your skeletal muscle are present here myofibrils like this. Myofibrils are present like this. Okay, these myofibrils, they have alternative light and dark bands. So if the information through neuron is sent to the myofibrils, only then the muscle will contract, because these alternative light and dark bands are made up of contractile protein. If only the contractile protein gets the information, only then the contraction will take place. Okay, so these T-tubules are present. If any neuron comes and innervate the muscle, it cannot send the signal to the myofibrils directly. So if it sends it to the T-tubule is present just adjacent to the myofibril, it can easily send the information here, right? So these are your neurons. What are these? Neurons. And what are these? Myofibril. And myofibril have alternative light and dark bands. These are alternative light and dark bands. And what are they made up of? Contractile protein. Alternative light and dark bands. And these position of alternative light and dark bands forms a structure here known as sarcomere. What is a sarcomere? We'll talk about that very shortly. But before that, let's see, these myofibrils can also move from their position. So to hold them into position, there is one protein which will join these myofibril to the sarcolemma. And what's the name of this protein? The name of the protein is dystrophin. The name of the protein is dystrophin. Apart from that, it also have these structure known as sarcoplasmic reticulum. These sarcoplasmic reticulum stores calcium ions. What does this store? Calcium ion. So sarcoplasmic reticulum is basically modified. It is modified smooth endoplasmic reticulum. It is modified smooth endoplasmic reticulum that can store calcium ions now. Why calcium? Because calcium plays a significant role in the muscle contraction, and I'll let you know again shortly in the next topics of the chapter. Okay, so you must be thinking, ma'am, you always say very shortly, very shortly. See, the things go in sequence. They are always right. All right, guys, so that's the structure of a muscle cell, skeletal muscle cell. Now let's see what is this sarcomere. Let's talk about the sarcomere or the structure of sarcomere. This is very important, so listen to me very carefully. Sarcomere is the structural and functional unit of skeletal muscle. Structural and functional unit. So this sarcomere is only present in cardiac muscle and skeletal muscle. It is absent in the smooth muscle. It is absent in the smooth muscle. So why this sarcomere has been formed? This sarcomere has been formed due to a reason of presence of alternative light and dark bands. So this is how alternative light and dark bands they look like. Alternative light and dark bands are formed of myofilaments. What are they made up of? Myofilaments. Myo means muscle. Filaments. These are containing contractile proteins. Okay, just like we say, cytoskeletal elements are present. You must have heard of them in this cell chapter, actin and myosin. Okay, so these your alternative light and dark bands, they are formed by, we say, okay, let's go here. They are formed by alternative light and dark bands. The light band is formed by the actin, troponin and tropomyosin. What are these, guys? These are myofilaments, right? Whereas dark band is formed by myosin. It is formed by myosin. Okay, so this is light in color because the band which is formed by this one is very thin. It's very thin, so it will appear light in color. Anything which is broader and darker or thicker, it will always appear dark in color. Okay, anything that is thicker will be dark in color, and anything which is thin will be light in color. So this one, they together form thin myofilament. So we say that your light band which is formed here, it is basically made up of thin filament. And what does thin filament have? These three proteins. What are these proteins? Okay, similarly, this black color, which are the spaces I've left, these are dark bands. They are formed by myosin, and myosin is forming thick filament, thick myofilament. Okay, thin and thick is just on the basis of how do they appear. If they in appearance they are thin bands. If in appearance they are thicker, that will be thick band. Okay, so these structures, they are forming the structure known as sarcomere. Okay, all right, let's see. For example, this is a thin filament. What is this, guys? Thin myofilament or thin filament. This thin filament, as I've already told you, this will form your light band. Which band? Light band. And light band and this thin filament, it contains the protein actin, troponin, and tropomyosin. Okay, we'll also study the structure of these proteins. All right. Now inside is a dark band, and this dark band is made up of which filament? It is made up of thick filament. And thick filament is made up of, yes, you all know that now. It is made up of your myosin. Okay, so this is the thick filament. And it is made up of myosin. All right. Now, there is a thin line of proteins. What? There is a thin line of protein that is bisecting your light band or your thin filament, and the name of that line is Z line. So there is a line that is bisecting. There is a line that is bisecting your thin filament. You call it as Z line. If you'll see in this structure, there will be a line that will be bisecting these. So this is forming section. The small unit has been formed, and this one unit will be known as sarcomere. Okay, similarly here, the area between two Z line is 1 unit. Similarly, there will be another line that will be the different unit. So the area between the two Z line, you call it as sarcomere. So we have divided the entire area into bands. Okay, so any band, okay, listen to me very carefully, this is quite serious here. Any band which have only light filament, or we can say only thin filament, only light band here, this portion, you will call it as I band. What you will call it as? I band. Okay, do you understand this? This type of a band, this portion has only white color filaments. This portion also have only white color filament. So you call it as I band. Now there is a portion, guys, which have both thin and thick filament. You call it as A band. What you call it as? A band. You can see here, there is also some portion of white here, and there is also portion of thick filament. So that is a A band. So A band's full form is anisotropic. Iso means similar, but it is not made up of similar units, so it is anisotropic. Isotropic, I band is isotropic. I stands for isotropic. Iso means similar. It is made up of only one thing, and that is all are same. So you call it as an isotropic band. Okay, in the anisotropic or A band, it is made up of both thick and thin filament. Both thick and thin filament, whereas I band is I band is isotropic band, and this band only contains thin filament. Okay, now there is a space, as you can see, present between, there is a space present between two thin filament. You call it as H zone. What do you call it as? H zone. And H zone is also bisected. H zone is also bisected by some proteins, and that line is known as M line. Okay, this space between two thin filament, this is your H zone. What is it? H zone. And it is also have been bisected by a line, M line. What are these lines? These are not the lines drawn by the pen. These are the lines of proteins. There are certain proteins present in a particular fashion. Okay, so this sarcomere, it is made up of this half I band, this half I band, and this complete A band. Why this is half? Because half of this I band is present on the subsequent sarcomere. So sarcomere is particularly made up of half I plus full A plus half I band. This is how it is made up of. Okay, so this is the structural and functional unit of your muscle. So this is all about your skeletal muscle and a very important structure known as sarcomere. Okay, guys. All right, let's talk about the smooth muscle now. The another name of smooth muscle is visceral muscle or unstriped or non-striated? Okay, why this is smooth? Why this is smooth? Because it has smooth texture. Because in this, no alternative light and dark bands are present. No light and dark bands present. But it doesn't mean doesn't mean that this will not be having the myofilaments. It's a muscle. Definitely it will be having myofilaments, but the arrangement will not be like that. As such, the alternative light and dark bands will not be present. Myofilaments will be present. If there are no myofilaments, that means no contractile protein, then it is no more a muscle. Okay, so no light and dark bands are present. Visceral muscle, because it is present in visceral organs. Present in visceral organs. Now, what are the organs where it is present? Like your gastrointestinal tract, reproductive tract, in your blood vessels. Right? What's the purpose there? It will always lead to the movement of things. Like in GIT, movement of food. Like in reproductive tract, movement of gametes. Like in the blood vessels, obviously the pumping action of the blood leads to the movement of blood. Okay. Then this muscle is involuntary. This muscle is involuntary. That means it is not under your control. Why it is smooth? Because it has no alternative light and dark band. If you'll see the shape of this muscle, the shape is fusiform. What is the shape? Fusiform. What was the shape of the skeletal muscle? It was cylindrical. It is fusiform. Let's do the opposite things. The skeletal muscle was multinucleated, or it shows a syncytial condition. But here you will get only one nucleus in the center. So we say it is uninucleated. It has no alternative light and dark bands. That's why its texture is smooth. Smooth texture. Okay, for more elaboration, what's the fusiform shape? Where it is tapered at both the ends. Okay, now here the myofibrils will be present in the longitudinal manner like this. So these are myofibrils containing myofilaments. What is absent here? Sarcomere, sarcoplasm, and sarcolemma. All these are absent in this muscle. All these are absent in this muscle. But in these muscles, gap junctions can be present. Gap junctions can be present. For example, if there are two smooth muscles and they have gap junction in between, they can work as a single unit. Here, the information can be transferred, or nerve impulse can be transferred very easily. So they can work as a single unit if the gap junctions are present. One will get the neuronal signal, other will send it to the another one, and they will contract together as a single unit. So gap junctions are present in the single unit type of a muscle. Okay, all right. Let's talk about the cardiac muscles, guys. If we talk about the cardiac muscle, where it is present in heart. Is it voluntary or involuntary? Do you have any control on your heart? No. So it is involuntary. It is involuntary. That means it is not under your control. This muscle is cylindrical like that of skeletal muscle. It is cylindrical in shape. And just like that of the skeletal muscle, it will be having sarcomere, sarcoplasm, and sarcolemma. If I say it has sarcomere, definitely it will be having alternative light and dark bands. So difference is the alternative light and dark bands in the cardiac muscle, they're very faint as comparison to the skeletal one. The reason is the cardiac muscle, they are very thick. So if you have anything thick, the bands will not be very visible. Okay, so here faint light and dark bands present. Faint light and dark bands are present. Now just like that of smooth muscle, it is uninucleated. It is uninucleated. And also just like that of smooth muscle, it is involuntary. And also one important point, your your cardiac muscle, these are branched. So this is an exclusive feature of this muscle, that these muscles are branched. As you can see these bridges, oblique bridges. Can you see them? I can see them. These are oblique bridges. oblique bridges are present, and hence we say that your muscle is a branch muscle. This is a very common question asked. Is the cardiac muscle striated or not? Yes, it is striated. It have alternative light and dark bands, but they are faint. Second, what's the shape? It's cylindrical. Third, is it branched? Yes, totally, this is branch. Apart from that, between two cells are present these discs known as intercalated disc. Intercalated disc. So these intercalated disc, this year a very smart question was asked, very clever question. Which of the following are present between two vertebra? In between two vertebra are present intervertebral disc, and they have also given the option cartilage, but in place of intervertebral disc they have given intercalated disc. So see how smart. So sometimes you get confused between these two terms, intercalated and intervertebral. So don't get confused. So intercalated disc are present between two cardiac muscle cell. These have two type of a junction. One is a gap junction, another a desmosome. Okay, so that's your cardiac muscle, guys. Let's talk about the structure of the contractile proteins. So the contractile proteins are there are two types of contractile proteins, or we can say there are two types of myofilaments. One is a thin, another is a thick. Okay, you have two types of myofilament. One is a thin, another is a thick. Now, what's the difference between them? How they work? What's the structure? How the muscle contract? We will discuss all about that. And it's also kind of an important. So you have to study it really well with me. So let's get started. So now what these thick and thin filaments they are made up of. So as I've already told you, these filaments are responsible for the dark and light bands formation, and also these thick or thin myofilaments, they are made up of proteins which are contractile proteins, the myosin and actin. So thin filament is made up of actin, troponin, tropomyosin, whereas your thick filament is made up of myosin. So let's talk about them in more detail so that you can get to know what actually they are made up of and how do they look like. And yes, guys, this is very important. From here, the most important parts of this chapter starts, from where, often the questions are asked, right? One type of a question can be asked from like what are the various types of muscles. The muscles revision we have already done, so it was a kind of a revision. Most of the part we have done in the structural organization in animal. One or two things can be new there, and other things we have already covered in the structural organization. So for all those who does not have attended my lecture of structural organization in animals, first go there, so read and learn the muscle part, and then come back and see what it is, right? So anyway, so let's get started with the structure of contractile protein. So guys, we have two types of myofilament. One is a thick, another is thin. The thick myofilament is also known as primary myofilament because it is performing major role. This is the most important one, right? So we'll start with the primary myofilament first. Thick myofilament, also known as primary myofilament. Primary myofilament. This is primary because this is important. This is performing very important function. So if you remember the structure of sarcomere, this was Z line. These were light bands, and these are basically the thin filament. And in the center were present the thick filament. So these are what? Thick filament. Okay. So we are going to talk about these one which I have drawn in the pink. Okay. So these are what? Your thick filament. So thick filament. Alright. These thick filament, they are made up of protein known as meromyosin. What they are made up of? These are made up of protein. And what is this protein? Meromyosin. This meromyosin is further made up of polypeptide chains. What they are made up of? Polypeptide chain. How many polypeptide chain? 6 polypeptide chain. What is a polypeptide chain? Your protein is made up of amino acid. And all the amino acid, when they join together, they form a peptide chain. What they form? They form a peptide chain. Okay. So here meromyosin, Here meromycin is made up of 6 polypeptide chain. 6 polypeptide chain. Out of these 6 polypeptide chain, 4 polypeptide chains, they are having low molecular weight. So you call them as 4, these are light chains. They have less molecular weight. Whereas 2 heavy chains are there. They have more weight, or they are heavier in weight. They have more molecular weight. Okay. Now, as I've told you, one meromycin is made up of 2 heavy chain. Let's draw it. So, for example, this is one heavy chain and this is the second heavy chain, like this. Okay. So, these chains, they are basically producing the entire structure of your meromycin. The light chain are present at this portion here, like this. 4 light chains. Okay, so this green one, these are the heavy chain. These orange ones, these are the light chains. Okay, so green ones are what here, guys? These are heavy chain. And these are the light chains. Now the entire structure has been divided into 2 portions. As you can see, this portion is the tail. This portion is the short arm, whereas this portion is the head. This is the head. Okay, so the entire body of meromycin is divided into 3 portions: head, short arm, and tail. Okay, so as you can see, up to this portion is your tail portion is present, and here short arm and head is present. So this portion is known as light meromycin, because its molecular weight is comparatively less. Light meromycin. Now you must be wondering, ma'am, but this one looks bigger. See guys, it already have 4 chains, 4 light chain, and then half of the heavy chain portion. So basically this portion will become the heavier in molecular weight or in the weight. So that's why this is known as heavy meromycin. This one is known as light meromycin. In short form, you call it as LMM. So a basic question can be asked. The light meromycin contains tail, short arm, head. You will say tail. Whereas short arm and head, they together forms heavy meromycin. They together form heavy meromycin. Fine. This heavy meromycin, what it contains? It is made up of short arm plus head. Now this heavy meromycin, sometimes it is present in a projected manner like this. Or if I show you here, the head and short arm emerges out like this from the thick filament. This structure is known as cross arm. What do you call it as? Cross arm. So this structure is cross arm. So what is cross arm? Basically, the head and short arm sometimes gets projected outside or towards outside. You call it as cross arm. You must have seen this diagram in the NCERT. Let me show you this diagram, guys. This one. So you must have seen this diagram in the NCERT. You can see here, this portion is the tail portion. This is light meromycin, whereas this portion with short arm and head is your heavy meromycin. So you can see short arm and head, they are projected outward. You call it as cross arm. Okay. Now, the head have 2 binding site. One is actin binding site, where the protein actin will bind, and second ATP binding site, where ATP, adenosine triphosphate, the energy currency will bind. So we say that our head, not our head, the myosin's head contain 2 binding site. One where actin bind, second where ATP binds. Okay. All right. So let's move back to this point. So the head, okay. All right, this one. So this head, it contains 2 binding sites. Let me write it here. Head contains 2 binding sites. One is actin binding site, where the actin will bind, and another is ATP binding site. ATP binding site. where the ATP will bind. What is ATP? Energy currency. Why do we need ATP here? We need ATP because we want muscle contraction. We want to move things, and for that definitely energy will be used. Okay guys, so that's about the primary filament. Let's talk about the secondary filament, or generally you call it as thin filament. So this thin filament is producing the light band. Which band? Light band. And this thin filament is made up of three types of protein: actin, troponin, and tropomyosin. Okay, let me show you how all they look like. So actin is basically, it is made by the polymerization of G-actin. G-actin, G stands for globular. This is a monomer. So G-actin are small molecule. When they polymerize in the presence of magnesium ion, they form F-actin. F means filamentous, like a fiber. So this is F-actin. So F-actin is the one which is present inside your thin filament. Okay. Also, this G-actin molecule have binding site here. Just like myosin have actin binding site, just like that actin have myosin binding site. Which binding site? Myosin binding site, because they both will join to each other. They are lovers, right? Actin and myosin, they both are lovers. I'll tell you a love story today. Okay, don't tell this to anyone. Keep it a secret. Okay. So here, troponin. Troponin is basically made up of three units: troponin T, troponin C, and troponin I. Troponin T, troponin C, and troponin I. Troponin T is the one which binds to tropomyosin. So that's how its name is troponin T, because it binds to tropomyosin. Whereas troponin C, in troponin C, C stands for calcium ion because it binds to calcium ions. And I is the inhibitor one because it is the one which mask means cover, to cover, right? It masks this side. Which side? Myosin binding site on actin. This is a villain. Like in every love story, there is always one villain who wants to have the heroine. But heroine and hero, they love each other. Who is the hero and heroine? Actin is the heroine. Myosin is the hero. Whereas troponin, especially the troponin I, this is the villain. So this will make sure that actin and myosin will not join, right? They will not join. They will not live together. So here, the G-actin's myosin binding site is always covered by the this troponin I during relaxation state. Whenever the muscle is relaxing, this troponin's component troponin I will mask or cover the myosin binding site on actin, right? Whereas tropomyosin, this is also filamentous and this is also double helical like this, just like that of actin. Actin is also double stranded. Two strands are there. So a common question can be asked: the two strands of actin and two strands of tropomyosin are present in your thin filament, guys, in your thin filament. Okay, let me show you how they are arranged. Okay, so for example, this is the actin. Okay, this is the filamentous actin, F-actin, present like this. Two strands, double helical. Okay, on this are present the strands of tropomyosin. Imagine this is also double helical. I've just drawn one, but imagine they're also double helical. Okay, so these are also double helical, tropomyosin. And these are also double helical. Double means two. There are two strands and both are helical. Next, on these are present three molecules of your of your troponin. Imagine this one binds to the tropomyosin. So this is troponin T. This one is binding to the actin and masking the myosin binding site. So this one will be I, and this one is the C. This will bind to calcium ion. So a lot of these subunits of troponin you can find on the, after some interval in this thin myofilament structure. Okay, let me show you here the diagram of NCERT. You can see in this diagram, after some interval, the troponins are repeating. So there are a number of troponin subunits present on one strand of thin filament. Okay, guys? Okay, all right. Okay, so next we have is this structure. Let's do some questions out of this structure. The light band contain actin and is called first I band, A band, H zone, Z line. Do it. Yes, my voice is a little different today because I am having a little cold, guys. Okay, so it's not the cold, it's like, sometimes there is so cold outside and you're not having such kind of a cold with mucus, but just your voice is little, it settles down like sore throat or something. So it's just how I feel today. Okay, I hope you can understand that a little. All right, so the light bands contain actin and it is called I band. Whereas if it have talked about the dark band, then you can also mark A band there. Okay, next question. Each organized skeletal muscle in our body is made of a number of muscle bundles or fascicle held together by a common collagenous connective tissue layer called. So what is that connective tissue which is holding all the muscle bundles together, guys? That's fascia. So answer is 3. Now what is the epimysin, perimysin and endomysin? Let me explain you. Though it's not given in the NCERT, but for your knowledge I can tell you about these also. So the entire skeletal muscle is covered by a connective tissue like this. This is known as epimysium. What do you call it as? Epimysium. Okay. Around muscle bundles, also there is one connective tissue present which is surrounding these muscle bundles like that. This is perimysium. Whereas around these muscle cells are also present some collagenous connective tissue, and that is endomysium. How it is different from the fascia? Fascia is the one which is present between the spaces, whereas perimysium is the one which is present around each muscle bundle like this. Okay, so do not get confused. If they're asking you in the question that which is the one which is holding all the muscle bundles together, then that will be your straight away fascia. Okay, and if they are asking which is the one present around each muscle bundle, then you will write perimysium. Okay, all right. Next, fusiform. Okay, fusiform, uninucleated and involuntary. Which is this muscle, guys? Smooth muscle. So smooth muscles, another name is visceral muscle. So answer to this question will be 4. Clear na? All right. Next. In the resting state, a subunit of dash masks the active binding sites for myosin on the actin filament. Very straightforward question, line from the NCERT, and that is troponin. So the component of troponin, which troponin? Troponin I, mask the myosin binding site. Okay, all right. Next, the heavy meromycin is formed of, you all know, short arm plus head, whereas the light meromycin is containing only tail. Okay, next, mechanism of muscle contraction. So these we have already done. Okay, these structures, how do they look like? What are these? The thin and the thick myofilaments. We have already done these. But now how the muscle contraction takes place? That's the question. How are my muscles are contracting? Like I'm doing this, this and that. You know, even if I dance like that. So why? How I'm doing that? So I'm doing because my brain is giving signal. My brain is giving signal, only then I'm dancing. Okay, for example, if you want to do some dance step like this, So my brain sent a process, my brain sent an impulse. Only then this muscle contraction it took place. Okay, now I'm not going to dance more for you. So here, the muscle contraction starts from the signal from the neuron. Imagine guys, this is a neuron. This is a neuron and these are the axon of a neuron. This is what we have done. And what type of a neuron this is? This is a motor neuron. What type of a neuron this is? This is a motor neuron. So motor neuron is innervating your muscle fibers. One motor neuron can innervate a number of muscle fibers and you call it as motor unit. What do you call it as? Motor unit. As you can see here guys, one motor neuron is innervating number of muscle fibers or muscle cells. So this type of a structure is known as motor unit. What do you call it as? Motor unit. Okay, we'll also study the NCERT of it, don't worry. So one motor neuron innervating a number of muscle fiber is known as a motor unit. Now, if you'll zoom this out, this portion where a neuron is innervating a muscle cell, this portion where a neuron is innervating a muscle cell, you call this as neuromuscular junction. What do you call it as? Neuromuscular junction or motor end plate. What's the difference between motor end plate, neuromuscular junction and motor unit? These two are same. Okay. The difference between neuromuscular junction and motor unit is in this entire structure where one motor neuron is innervating number of muscle fiber. Whereas the point where muscle and neurons are joining to each other, you call it as neuromuscular junction. Let's draw a neuromuscular junction, guys. Okay. Let's draw a neuromuscular junction. So imagine this is a neuron. This is a neuron. And here you have skeletal muscle fibers with the T-tubules like this. Okay. So for example, how do the muscle contractions start? You need to understand the structure side by side. And I will also be telling you the functioning or how does it work. So here guys, first of all, the impulse will arise, nerve impulse. Okay, nerve impulse will come here. And this is the axonal end of motor neuron. Axonal end of motor neuron. Alright, so at the axonal ends or the synaptic bulbs are present synaptic vesicle. At the ends of the ends of the axon, they are known as synaptic knobs or axonal end bulbs. So this structure is axonal end bulbs or synaptic knob, and inside that is present synaptic vesicles. And these synaptic vesicles, they contain neurotransmitter. What do they contain, guys? Neurotransmitter. Okay, so this is synaptic vesicle. And inside them are present neurotransmitter. Neurotransmitter are nothing but chemicals. What are these? These are chemicals. Let me give you an example like acetylcholine. Acetylcholine's full form is its short form is ACH, acetylcholine. Okay. Now what happened here, when the nerve impulse arises in the neuron, these synaptic vesicles, they will fuse with the membrane of axon. They will fuse with the membrane of axon and they will release its neurotransmitter. And the process by which they release the neurotransmitter is known as exocytosis. So once they release the neurotransmitter, they are going to bind with these channels on the muscles. So imagine they bind to these channels and these channels will open and it will lead to the entry of sodium ions. So when the sodium ions enter inside, the muscle cytoplasm, this causes muscle excitation. What does this causes? This causes muscle excitation, also known as depolarization, just like we have done in the cardiac muscle. So what happens when the muscle get excited? when the muscle will get excited, what will happen? In the cytoplasm are present these sarcoplasmic reticulum. So this wave of excitation or depolarization will hit the sarcoplasmic reticulum. This will lead to the exit of calcium ions, and these calcium ions will come in the cytoplasm. So once these calcium ions, it enters inside the cytoplasm, these calcium ions will bind to troponin C. You heard it right. They will bind to troponin C. Okay, let me show you now. All right. So they will bind to this troponin C. These calcium ions, they will bind to this troponin C. So, okay, let me just repeat it one more time so that you can catch things up, guys. So what happened when the nerve impulse arises from the neuron, it will lead to the release of these chemicals from the neurons known as neurotransmitter. These neurotransmitter will bind to these sodium ion channels. What are these? Sodium ion channels. So when these chemicals or acetylcholine, they bind to these sodium ion channels, sodiums enter inside the muscle, causing the muscle depolarization. Always the muscle gets excited whenever there is or depolarization always occur in muscle or a neuron whenever the sodium ions enter inside the muscle or a neuron. Okay, so this wave will hit the sarcoplasmic reticulum, and sarcoplasmic reticulum, who already have a lot of calcium ion in it, it will release the calcium, and calcium concentration in the cytoplasm of muscle will increase. Okay, now this calcium ions concentration has been increased in the cytoplasm of the muscle. Now what will happen? These calcium ion will bind to troponin C. Okay, now what will happen? Calcium ion binds to troponin C. They will bind to troponin C. Once they bind the troponin C's conformation will change, or it will start moving just like this. It will start moving all these components, and they will come into position like this. Like this is troponin I, T and C. It will move all the components in this direction, and hence they will straighten up. And you can see here the myosin binding site is free. The myosin binding site is completely free. So when the calcium ion binds to troponin C, the conformation changes. And troponin I unmasks, or troponin I moves, unmasking myosin binding site. Unmasking myosin binding site. So when the villain is out, the troponin I was the villain of the story. So when the troponin I or the villain is out, the troponin I is out, now the actin and myosin can easily meet. Okay, so this is how your myosin binding site has been masked. But now still, muscle contraction is still a mystery. Half of this is done. The signal has been sent to the muscle. Now how the muscle will contract? Let's see. So there is a theory behind muscle contraction, guys, and the name of the theory is sliding filament theory. What's the name of the theory? Sliding filament theory. So let's see what happened in a sliding filament theory. So let's get started. Sliding filament theory. Okay, now as you all know, this is your thin filament. I'm just drawing one part of a sarcomere, not the complete sarcomere. And this is your thick filament myosin, and this is the cross arm of the myosin. Okay, this is a cross arm of the myosin. This is the thin filament. For your understanding, let me write thin filament, and this is thick filament. Okay, and as you know, the ATP always bind to the head of the thick filament. This is how ATP binds. Okay. Now when the ATP breaks out, it will give energy. It will give energy. So imagine in the second step, what happened in the second step? The ATP breaks down. And when the ATP breaks down, it forms ADP and IP. Okay, this is what happened. I'm just zooming out. I have made the bigger one because that will be congested for you. So what happened? The ATP here, imagine the calcium has been bound to the troponin C. Conformation changes. Myosin binding site have been unmasked. Now all tasks will be done by the myosin. Now myosin have ATP on its head, and that ATP will be utilized. When the ATP breaks down, what happened? When the ATP breaks down, this thin filament will bind to, or this head will move up and it will bind to the thin filament. The head will move up and bind to the thin filament. Okay. Here IP has been released. That's why when the inorganic phosphate gets released from the ATP, only then it can give the energy. So when it gives the energy, the head got the energy to get bound to the thin filament. Okay. Now what will happen next? Now ADP is already attached here, IP has been released. Now ADP will also release. ADP will also release. So when ADP gets released, the conformation changes occur. What occurs? Conformation changes in the head. Head will change its position. As you can see, earlier head was like that. Now when the ADP gets released, head will move in this direction. Okay. So when head has changed its direction, at this point, it will slide the thin filaments towards inside. It will slide the thin filaments towards inside, and there will be a movement. So you call it as power stroke. You call it as power stroke. Okay. Now when new ATP will add, or new ATP is added to the head again, the conformation will move back to its original position and the thin and thick filament will separate and ready for the next cycle. Okay, let me make it very clear for you. I know in one go it's very difficult for you to understand in one go. Okay, so for example, this is the myosin, this is the thin filament. Right now ATP has been attached. Right? Okay, let me have some things for you. For example, this is ADP, this is IP, and together they are forming ATP. Right? This is the head is attached to ATP, and this is the thin filament. Okay, now if the myosin head has to bind to the thin filament, it has to break into ADP and IP. And when IP will be released, and ADP is already attached, the head will get the energy and it will bind to this one. Okay, right? Now when ADP will be released, imagine ADP is released, this head will be moved in this direction and leading to the movement of thin filaments towards inside, like that. Okay, so basically if I have this sarcomere, okay, here we have thin filament and this is a thick filament. So these heads are attached like this. So they will move the thin filaments towards the inside, And this is how the thickness of the sarcomere will reduce during contraction. So this is when the contraction has been started. Okay. Now when the further contraction will take place, they are moving more towards the center like this. They are contracting. They are moving more towards the center like this. Okay, and in the place when maximum contraction take place, these thin filament will overlap each other like this. These thin filament will overlap each other, and this is how thick filament will be present. So this is one when it is normal and it has started contraction. Here it is contracting, and here maximum contraction. So one thing that you can note here, guys, is during muscle contraction, these filaments are moving towards the center. First point, they are moving towards the center, right? Second point to note here is that when it is contracting, the length of the fibers, it remains the same. Can you see anywhere the length has been reduced? No, the length remains the same. There is only overlapping of the filaments. Second point to note here is that the I band disappears during maximum contraction. What was the I band? This portion was the I band. This portion was the I band. Right? So can you see any I band here? No, only A band is present here. So we say that during maximum contraction, I band disappears. I band disappears. Second, A band remains constant. A band remains constant. Okay, and also during contraction, length of myofibrils, length of the myofilaments remains same. As you can see, no length has been changed, only the filaments are present towards each other. For example, you are sitting in a class and there is a row of tables, right? And there are 3 rows, 1 2 3. Okay, so when there are 3 rows of tables in the class, in the center there is a space present. Okay, what if I join all the 3 rows together? The center place will disappear. All the tables are joined together. So during this process, is there any change of the length of tables? No, the length of tables remain the same. Only they have been joined to each other. Just like that, myofilaments, they're just overlapping each other. They have not changed in length so far. Okay, so let me just pick up my ADP. Okay, all right. So let's see this diagram of NCRT. In this, you can see this is a muscle when it is in a relaxed state. This is the length of a sarcomere here. This is the length of a sarcomere here when it is in the relaxing phase. Okay, when it started contracting, you can see the length difference. This middle line is constant. They have just reduced this one to tell you that the sarcomere's widthness is decreasing. Okay, so in the maximum contracted state, the this width of the sarcomere, it has reduced as much as it can. Also, you can see I band is present here, I band is present here, I band is present here, but the size have been reduced, and here no I band is present, but A band remains the constant in all the places. Also here, during maximum contraction, you can also notice one more thing. H zone is visible over here. Here H zone has been reduced, but here H zone also disappears. So we can also write this point that during maximum contraction, H zone disappears. H zone disappears. Okay, all right. Let's talk about NCRT. All right. So muscle contraction is initiated by a signal sent by the central nervous system via motor neuron. So as I also told you that the signal is sent towards the muscle through the central nervous system, especially the brain. And what neurons are bringing signal towards the muscles? Motor one. Motor is the one that is related with some movement. Okay, so what is moving here? The muscle. So always motor neurons innervate your muscle, especially skeletal muscles. Okay, next, a motor neuron along with the muscle fibers connected to its constitute a motor unit. So what is a motor unit, guys? Okay, I hope you all have opened your NCRT books, if I'm not wrong. Okay, so these are connected to the muscle fiber. You call it as a motor unit. The junction between a motor neuron and the sarcolemma of the muscle fiber is called the neuromuscular junction. So that union of the muscle with the neuron, what do you call it as? Neuromuscular junction. And what part is attaching basically? The sarcolemma. If you have seen this structure, guys, if you have seen this structure, then what is this part which is attached towards the motor neuron? What is this portion? This is sarcolemma. This is sarcolemma, right? And what are these? These are acetylcholine. And this red is a sarcolemma. Okay, all right. Anyways, a neural signal reaching this junction leaves a neurotransmitter acetylcholine, which generates an action potential in the sarcolemma. So once the nerve impulse comes or it reaches towards the muscle fibers, then it will start contracting. And how it will contract? Let's see. This spreads, or it will release a neurotransmitter. And what's the name of a neurotransmitter? They have given acetylcholine. Okay, this spreads through muscle fiber and causes the release of calcium ion into the sarcoplasm. Now that depolarization wave, it will go to what? It will go to your sarcoplasmic reticulum, and that will release the calcium ion in the cytoplasm. And what is the cytoplasm of muscle known as? Sarcoplasm. Increase in calcium level lead to the binding of calcium with the subunit of troponin on actin filament, and thereby remove the masking of active site for myosin. So it is saying the calcium ion will lead to the binding of calcium with subunit of troponin. Once the calcium binds to troponin C, it will move all the troponins and there will be unmasking. Utilizing the energy from ATP hydrolysis. Now ATP hydrolysis occur. What is ATP hydrolysis? Breakdown of ATP. The myosin head now binds to the exposed active site on active actin to form a cross bridge. Now myosin head will bind to the myosin binding site on the actin, and what will be formed there? A bridge will be formed there. And what do you call that bridge? Cross bridge. So this is the diagram which is given in the NCERT. So this portion that will be formed here, guys, this portion that will be formed here where thin and thick filaments are attached, you call it as cross bridge. How cross bridge is different from the cross arm? How cross bridge is different from the cross arm? This portion which is projected outward is cross arm. When the cross arm binds to the thin filament, you call it as cross bridge. So it's a bridge between thick and thin. Okay. Now, this is what myosin or the sliding filament theories diagram that NCERT has have given. So first of all, there is ATP, right? There is ATP which will start from this point. ATP that has been attached to the myosin head. This ATP is going to break down into ADP and IP. Now when the IP or inorganic phosphate, it is released, the myosin head will bind to the actin and the cross bridge will be formed. Once this cross bridge is formed and ADP is released, you can see here ADP is also have been released here, the head will move. The head will move, causing the sliding or rotation. So once the sliding and rotation has taken place, a new ATP will bind, which will break the cross bridge, Okay. So very simple. Here ATP breaks down into ADP and IP. IP release, attachment. ADP released, sliding. ATP attachment, detachment. So always on binding of new ATP, there will be detachment. You have to write this line in the notebook also. Okay. So you have to write this line that during attachment of new ATP, there is breaking of cross bridge. Okay. Yes. I hope that's pretty clear to everyone now. I have explained you thrice this point. Okay, guys. Now, okay, so I missed the word here, I guess. This pulls the attached actin filaments towards the center. As I'll say, it will always move towards inside of A band. The Z line attached to these actins are also pulled towards inward, thereby causing a shortening of the sarcomere, that is contraction. So as I've told you, always the width of sarcomere will decrease. You call it as shortening of sarcomere as well in the English, in another words, okay? And you call it as contraction. Now, what about relaxation? So do not read the next lines because that's about relaxation. So let's get started with the relaxation part, guys. What about the relaxation? Contraction, okay, you know about contraction now. You know about the contraction now. What about the, okay, what about the relaxation? How does relaxation takes place? Just opposite to contraction. So if you remember, how did the contraction started? Contraction started when the nerve impulse arises in the motor neuron. When the nerve impulse arises in the motor neuron, it releases chemical which will causes the excitation of muscle, and that excitation causes the opening of sarcoplasmic reticulum, which have a lot of calcium ion. So the entire purpose of this was to release calcium ions from the sarcoplasmic reticulum. Right? So the entire purpose of this neuromuscular junctions, whatever it is happening, is to release calcium ion from the sarcoplasmic reticulum. What if the nerve impulse stops? The muscle contraction will also stop. Okay? Muscle contraction will also stop. So how does relaxation start? Nerve impulse stops first of all. What will stop, guys? Nerve impulse. Nerve impulse stops. So when the nerve impulse stops, there will be no acetylcholine release. When there is no acetylcholine release, there will be no excitation of muscle. No excitation of muscle and no release of calcium ion from sarcoplasmic reticulum. Okay, now what if we want to stop the muscle contraction, but there is already some calcium ion present in the cytoplasm? Okay, if you don't get it, listen. Imagine this is a muscle cell and these are sarcoplasmic reticulum. During first contraction or first nerve impulse, this has released some calcium ions. But immediately the nerve impulse stops and we want to stop the muscle contraction. But calcium ion is already present inside the cytoplasm. Then in that case, if calcium ion is already present in the cytoplasm, but we don't want the muscle to contract, this sarcoplasmic reticulum will take the calcium ions back. You heard it right. So sarcoplasmic reticulum can also take back the calcium ions. So calcium ions in the cytoplasm are taken back by the pumps. What are the pumps? Pumps are certain channels that will use one ATP and take calcium ions back, right? So they are taken back by the pumps of sarcoplasmic reticulum. Sarcoplasmic reticulum. Now you know sarcoplasmic reticulum is a smooth endoplasmic reticulum, is a modified smooth endoplasmic reticulum. So it has a structure known as cisternae. So that cisternae are the one that basically have pumps and take the calcium ion back. Okay, that's about the relaxation. This is how the muscle relax. Contraction, you know, the impulse will be sent by the CNS to the your muscles. But how do the relaxation takes place? Now you know that too. Okay, so guys, read the NCERT with me. Open your NCERT. It is clear from the above steps that during shortening of the muscle, that is contraction, the I bands get reduced, whereas A band retain the length. This is clear. During maximum contraction, the I band disappear and A band remains constant. The myosin releasing the ADP and PI goes back to its relaxed state. A new ATP binds and the cross bridge is broken. So when the new ATP binds, the cross bridge gets broken down. The ATP is again hydrolyzed by the myosin head and the cycle of cross bridge formation, okay, that's what we have already seen, and breakage is repeated, causing further sliding. So when the new ATP will come, next cycle will start. For example, we started from here. First of all, IP will be released, attachment. ADP release, movement. New ATP come, detachment. Now, new ATP will hydrolyze, IP release, attachment. ADP release, sliding. New ATP come, detachment. Again, next cycle will start, ATP hydrolysis, attachment. Now, ADP release, sliding. New ATP come, detachment. So this is how you can see this is going more towards inside. Okay, all right. So this is how relaxation takes place. The contraction takes place. Now about relaxation. The process continues till the calcium ions are pumped back to the sarcoplasmic cisternae, resulting in the masking of actin filament. So how does relaxation takes place? When the calcium ion is no more available for binding to troponin C, and I will keep on masking the myosin binding site on your actin. Okay, this causes a return of Z line back to the original position, that is relaxation. So when the Z lines are moving towards inside, leading to shortening of sarcomere, what will you call it as? I'm asking you a question. If the Z lines are moving towards inside, leading to the shortening of sarcomere, what will you call it as? Contraction. If the Z lines are moving back to the original position, leading to the original size of the sarcomere, what it is known as? Relaxation. Okay. All right. Now let's talk about some other things about muscles, guys. The first thing that we're going to talk about is muscle fatigue. What is muscle fatigue? Fatigue means to get tired. You get tired. For example, if you do not do a lot of exercise and one fine day, I ask you to run continuously for 3 kilometers. What will happen the next day? You will get cramps. Right? So, or whenever you're doing hiking or going to certain temples by walking, like in Vaishno Devi, people used to, climb up the mountains and go to that temple. So when they return, they usually feel cramps in the muscles and they are not able to walk properly. So what are these cramps and what is muscle fatigue? First of all, muscle fatigue is a property of skeletal muscle. It's a property of skeletal muscle. That's a property of skeletal muscle. Property of skeletal muscle. Okay, I tell you one story of mine of muscle fatigue. when I was in my college, our university used to, organize those trips for us. So they were not organizing some great trips like Goa and so. So the first trip that came when I entered my university was of Vaishno Devi Temple and Golden Temple in Amritsar. So we were very excited, and it was of very low cost at that time. It was just of 800 rupees, and for us, spending a lot of money like thousands was not possible. So we said, "Yeah, that's 800. We'll arrange and we'll go." So some of our friends, I asked some of my friends, and they were also ready, and we went to that trip. And we were so much energetic. Yes, we'll go, we'll go. And when we reached and we started tracking, we used to, hike the mountain there in Vaishno Devi. And we were so much excited. And in excitement, we started running in our first 2 to 3 kilometers. And the first 2 to 3 kilometers are very much tough because our body is adapting there, right? And we reached the temple. We had a lot of nice environment there, had darshan, and everything was done. And when we moved back to our hostels, nobody was able to stand up properly, and we were not able to go to the in fact toilets. So this was a muscle fatigue, that was a muscle cramp. I felt that for the very first time in my life in that much quantity. Yeah, we usually get muscle cramp, we run or so, but those type of a muscle cramp I have never ever experienced in my life. I was barely able to walk for around 2 days, and at that time I realized that I will not do the same thing again. That was the stupidity we did because we were very young. And when you are young, you used to do a lot of stuffs like that. All right. So what happened to Diksha ma'am's muscle at that time? Let's see. So what happened when we do a lot of muscle activity? When there is a lot of muscle activity we are doing, and energy level is reducing, and your muscle is not getting enough oxygen as well, right? For example, if I say muscle activity is more in this point, I was hiking, I was running, right? Muscle activity was more. So when muscle activity is more, we need more ATP. But when we need more ATP, our muscle glycogen will start to break, and it will form glucose. This glucose, in the absence of oxygen, because at this point the oxygen is also less, in the absence of oxygen, this glucose, in the absence of oxygen, it will get converted into lactic acid. And when lactic acid got accumulated, this gives you cramps in the muscles, and you're not able to move your muscles. So this lactic acid, what will happen to this lactic acid? Later, after some time, maybe in a day or 2 or 3 or 4 days, depending upon how much lactic acid is accumulated, your lactic acid, with the help of blood, it starts to move and it goes to the liver. Okay, so this lactic acid, now it will move to the liver via blood. Now what will liver do? Liver will produce 80% of the lactic acid. It will convert this lactic acid into the glucose, and 20% of this lactic acid directly will be burnt, and it will form CO2 and water. Okay, so first of all, muscle activity was more, ATP requirement was also more, right? More ATP requirement. There is more ATP requirement. Not ATP is not more. We need more ATP. Okay. So requirement of ATP is more. So for producing ATP, we need to break the muscle glycogen and convert it into glucose. Now glucose, it has to burn or oxidize to give us energy. But there is not enough oxygen. When there is not enough oxygen, when glucose burns, it will form lactic acid. And lactic acid accumulation gives you cramp. And this is what muscle fatigue is. Now what will be the fate of lactic acid? This lactic acid will go to the liver, and liver will convert 80% of this into glucose, and 20% it will simply oxidize it, right? So now this glucose via blood, it will reach to the muscle, and it will become the part of the muscle glycogen. Since it is a cycle, so you call it as Cori's cycle. What do you call it as? Cori's cycle. So Cori's cycles explain about the muscle fatigue and the fate of lactic acid. So this was happened to me, and I was not able to move, but gradually after some time, this cramps, they went away, or they were gone because my liver took that and converted into glucose, right? So that's the fate of your lactic acid, and that's the story I have told to nobody but you guys. All right, so let's move further and talk about types of muscle fibers. Types of skeletal muscle fibers. Skeletal muscle fibers. It's very tough to write in capital sometimes. Your brain gets confused. So there are two types of muscle fibers, guys. One is a red muscle fiber, another is white. You heard me right? We have two types of skeletal muscle fiber. One is red, another is white. Let me drink some water. Okay. Now, what's the difference between two? Obviously, the color of the red will be red and white will be white. That I'm not going to discuss. Okay. First of all, why red is red? That can be a point of discussion. Because myoglobin pigment is more in red muscle fiber and less in white muscle fiber. Why red is red? Because it have myoglobin. Now you must be thinking, ma'am, we have not heard about myoglobin. Like hemoglobin is present in the blood and it carries oxygen. Just like that, it's the oxygen carrying pigment. Oxygen carrying pigment, especially in muscles. It's only present in muscles. So because always hemoglobin gives red color to the blood. Just like that, the myoglobin also gives red color. Okay, so the content of myoglobin is more in the red, but it's less in the white. Similarly, the blood vessels and mitochondria, they are also more, they both are more in the red muscle fibers, whereas they are less in the white. Okay, this means less, this means more. I will write this down here also. This means more, this means less. Okay, now sarcoplasmic reticulum. So sarcoplasmic reticulum stores, tell me, sarcoplasmic reticulum stores calcium ion. And anything that has calcium in it, what is the color of the thing which have calcium in it? Milk have a lot of calcium, it appears white. Bones have calcium, it also appears white. Just like that, white muscle fibers are white because they have more sarcoplasmic reticulum and they have less of it. Since they have a lot of mitochondria, they will always respire aerobically. They will always respire aerobically. They undergo aerobic respiration. So you call them as aerobic muscle fibers. And they always undergo anaerobic respiration. So you always call it as anaerobic muscle fiber. Let me give you an example. Your eye muscles, they are containing white muscle fibers, whereas your back muscles have red muscle fibers. They have red muscle fibers. Okay. Also, your red muscles are present in the flight muscles of the migratory birds, the birds that have to flew over the oceans to migrate from one place to another. So they cannot get fatigued. They cannot afford the anaerobic respiration. So they have red muscle fiber, whereas non-migratory birds muscle fibers, they have white muscles. Okay. So that's the difference between both the muscle fibers. So let's solve some question. I will only give you one question and you have to solve that. Let's get started. So let's do this question. Find the incorrect statement. You have to find the incorrect one. Anaerobic breakdown of glycogen causes muscle fatigue. This is absolutely correct. Whenever there is muscle fatigue, the glycogen breaks down anaerobically and it forms lactic acid. And accumulation of lactic acid causes muscle fatigue or cramps, right? Then cross bridge is broken down on the binding of new ATP. So every time a new ATP attaches to your myosin head, the bridge formation will be destroyed. Sarcoplasmic reticulum is more in aerobic muscle fibers. No, sarcoplasmic is more in white. And white, they are known as anaerobic. So the answer is 3. What is the fourth statement? Each thick filament is a polymerized protein. Very true. So each thick filament is made up of many myosin. How many myosin? Around 300 myosin or meromyosin. They form one thick filament. Okay, so the answer to this question will be 3, guys. Okay, all right. Next, let's start with the skeletal system. So if we talk about the human skeletal system comprises of 2 things. One are the bones, another is your cartilage. The difference between a bone and cartilage is that bone is quite harder because it have calcium salts. It is hard and it has calcium salts, right? Whereas the cartilage, they are pliable and they are containing not the calcium salts but chondroitin salt. Chondroitin, all right, chondroitin salts, right? As a result, it is not harder, just like that of bones. So usually you will find the cartilage in your joints. Where do you find them? In your joints. Okay. Now, if I talk about the bone and cartilage, what are they doing in our body? First of all, this is a part of your endoskeleton, so it will support the body. It supports the body. Second, they acting as a structural framework. Structural framework. Whenever you know, a building is made, at that time, if you have seen, those iron rods are usually inserted in the building to make a structural framework, and then on that the bricks and cements are applied, and that framework will give a rigidity to the and you know, strength to your building. In Hindi, you call those iron rods as sariya, right? So this is like your sariya, and it will provide the structural framework and it will support the body. Fine. And it also helps in locomotion and movement. It also helps in locomotion and movement. Now the entire skeleton is divided into two portions. I'm talking about bones. One, the present in the main axis. This one is your axial skeleton. The one which is present outside the axial skeleton is your appendicular skeleton. So we say that your human skeleton, which comprises of around how many bones? Around 206 bones in the adults, right? So adults have 206 bones. The embryo have more than 300 bones. Embryo have more than 300 bones, or around 300 bones, right? Whereas the human skeleton, the adult skeleton, like I, have around 206 bones, out of which the 80 bones are the part of axial skeleton and 126 bones are the part of appendicular skeleton. Now, what are the bones of axial skeleton? It's present in the main axis. Now, what are the various bones? Let's see. We have skull, ribs, sternum, hyoid bones, ear ossicles, and vertebral column. These are the various bones of your axial skeleton. Okay, let's imagine your body. You have everything, right? Skull, yes, move towards mouth. Below the tongue, you have hyoid bone. So hyoid bone is the bone which is present below the tongue. Okay, at the base of the tongue. This is just one bone, single bone. Then in ear, you have ear ossicle. So all these are the part of the skull, but skull forming bones are 22 bones, the one which are actually forming the skull. The one which are present inside the skull are the one of the ears, 6 bones, or they are present in 3 pairs, ear ossicles. Now, what are these ear ossicles? We have malleus, incus, and stapes. So, one in each ear, 3 here, 3 here. Okay, so total 3 pairs or 6 bones. Then come downward. Here you have in the this portion, you have sternum. You have a breastbone known as sternum, and then on the rib cage you have ribs, and at the back you have vertebral column. So the entire which are forming the rib cage. So rib cage is formed by the rib, sternum, and vertebral column. This we have already done in the chapter breathing and exchange of gases also. Okay, all right. So let's get started with the skull. So as I've told you, in skull, we have total like 22 bones, and these 22 bones, guys, out of this, the 8 bones are the cranial bones and 14 are facial bones. Cranial, which are forming this portion, facial, which are forming your face. An entire structure is a skull. Skull, khopdi, you have seen in the movies, right? So the cranium, this portion, cranium is also known as brain box. So cranial bones forms your cranium, and cranium is another name for brain box because here brain is present. Whereas the facial bones are these bones, right? So what are the cranial bones that we have? Let's get started. Frontal, parietal, temporal, sphenoid, ethmoid, and occipital. So how can you learn these? Few people try searching Every option. If you can learn it in this way, that's pretty good. See, here this is a diagram of a skull, but you don't need to because you have skull. Okay, first of all, we'll start with the frontal. Frontal is this one, which is forming your forehead. F for frontal, F for forehead. Then you have these temples. We say, no, oh, my temples are paining, this portion. These are temples. Again, these are two. Frontal, forehead is one. So one frontal, two parietal, right? Then we have not the two parietal, we have not done, two temporal. Then this here, you have parietal bones. Here you have parietal bones, okay? Then come the occipital. Occipital is one down here in this portion. So in these portion, you have these bones. What? Frontal in the forehead, parietal on the sides, temporal on the temples, and occipital here. Occipital here, okay? Now, let's talk about these two, sphenoid and ethmoid. Then I'll show you this structure. Okay, so you can see this is the skull, right? This is the skull, and here in skull, this one is frontal, this is parietal, this one you can see here, this is temporal, and this one is occipital, right? In the occipital, you can see this round structure. Can you see this round structure easily? Yes, I'll move it, then you can easily see this round structure, this circular structure, this opening. This one is known as foramen magnum. From here, the spinal cord comes out, right? You know, the brain is attached with the spinal cord. Medulla is the last part of your brain. It elongates to form spinal cord. So spinal cord comes out from this portion like this. This is the skull. Okay, please skull move. Yeah, okay. So this is how spinal cord will come down from this portion. Okay. Apart from this, occipital also have these projections, these spines, if you can see. Can you see these two spines? Yes, I hope you can see this. If not, there are two spines present, guys, there. Okay, let me just move it a little bit like that. Can you see this spine? Yes, this is the spine. These two spines are present and they are attached to your occipital bone, and they are known as occipital condyles. What are they? Occipital condyle, and they fit to the first vertebra of vertebral column. They fit to the first vertebra of the vertebral column. Okay, now let's talk about the sphenoid bone. Sphenoid bone, please skull move for me. Okay, so let me just check if you can see here. Okay, this is not a bit clearer, I guess. So if you can see this, this is a sphenoid bone present here. Sphenoid bone. It's a single bone present just in center of the skull. Just in the center of the skull. And sphenoid bone articulates. This one is a sphenoid bone. So this is only one bone present just in the center. It is forming the joint with the temporal, with the parietal and with the frontal. You can see the sphenoid. Yes. Let me show you the ethmoid now. Ethmoid is present just below the sphenoid. Here. Ethmoid is present here. This one is a ethmoid bone. This one is a ethmoid bone, right? So ethmoid is present just near to the sphenoid only. Okay, and then we have in the eye facial bones, and these all are your facial bones. You can see here, this is the orbit of the eye. Inside that is present the lacrimal bone. This one is a nasal bone which will form your nose, right? This part of a nose is basically made up of cartilage. That's why I can move it like this, right? So this one is a nasal. This one is lacrimal. Inside of that is ethmoid. All right. This bone is zygomatic. This is zygomatic, the cheek one. I smile and my cheeks are up. That means I have a bigger zygomatic bone. Okay. So this is a zygomatic. This one is mandible. This one is mandible, the single bone, and it moves. The only bone that moves here in the skull. This is maxilla, which forms the upper jaw, right? Okay, let's see. So I think all these bones are clear. Sphenoid is present just in the center, and below that is present ethmoid. Now, vomer bone, nasal concave, nasal, maxillae, mandible, palatine, zygomatic, lacrimal, all these bones which form your face. So you have seen the lacrimal, it's present in the eyeball, right? Zygomatic forming cheek. Palatine, where is palatine? Okay. So can you see this face? In the face, inside that, or just I'll show you a bone. Okay. You have tongue. You all have tongue, I guess, right? So put the tongue on the roof of your buccal cavity like this. Can you see that? So that is your palatine bone. Simple. So you do have palate, hard palate, and that contains a bone known as palatine. Now you have seen the mandible also, you have seen maxilla also, you have seen nasal also, vomer and nasal concave. So you have seen ethmoid bone, right? You have seen ethmoid bone. This is a ethmoid bone. So to ethmoid bone, if we can see, a number of bones are attached. So one such bone is your vomer bone. This portion is vomer bone, and on sides are present nasal concave. On sides are present nasal concave. So we can say that ethmoid, vomer, nasal concave, sphenoid. This is how they are arranged. Okay, you don't have to go in much of a detail. You just need to know only the names of them. Then how will you learn the names? Ma'am, again difficulty. No problem. Ma'am is there for rescue. Webber. Is there any Webber who is watching this lecture? Yes, if you are Webber, please comment. Webber can not. How can we say can? From concave. Make my pet zebra laugh. Ha 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 ha. Right. So this is how you can learn all the names. Especially the one basic thing that you need to know here is how many number of skull bones are there and what are their names and numbers, because it's actually mentioned in the NCERT. NCERT has given the diagram, so you should know which is a facial bone, which is a skull bone. Okay, guys? Okay. So let's talk about the occipital condyles and foramen magnum. So first of all, we'll talk about occipital condyle. What are occipital condyles? These are convex processes or spine-like processes that emerges out from occipital bone. From which bone? Occipital bone. Alright, so we are saying occipital condyles. That means they are 2 in number. Any skull which have 2 occipital condyle, you call it as dicondylic skull. You call it as dicondylic skull. Okay, second thing. Now, your occipital bone also have an opening known as foramen magnum. So occipital bone have opening. What's the name of the opening? Foramen magnum. Foramen magnum. And through foramen magnum, spinal cord emerges out. Spinal cord emerges out from here. Fine. What about sphenoid bone? One such bone is a sphenoid bone. In sphenoid bone, you have a seat for pituitary gland. This one have a seat for pituitary gland. For pituitary gland. And the seat has a name. Yes, so in the sphenoid bone, there is a small depression known as sella turcica. So on sella turcica is present, or pituitary gland sits on sella turcica, right? So that's about your skull, guys. Let's move further and talk about sternum. So the sternum is a dagger shape. Dagger means khanjar, right? It's a dagger shaped bone. Dagger shaped bone. It's a dagger shaped bone. It's one, single, just like you. It's single, one. This is present in just center of your ventral side of thorax. This is dorsal side, this is ventral side. This is dorsal side, this is ventral side. It's present in just in the center, right? So where it is present? Mid-ventral line. Mid-ventral line of thorax. So here I will not just explain you the sternum, rather I will explain you the entire rib cage. So if you remember, the rib cage was formed of 3 types of bones. One, the thoracic vertebrae. Thoracic vertebrae are the type of vertebrae of vertebral column. Second bones were ribs and third were sternum. Okay, so here I'll explain you everything about ribs, sternum and thoracic vertebrae. Okay, so if we'll talk about ribs, if I talk about ribs, ribs are S-shaped bones. S-shaped bones, and they are bicephalic. What are they? Bicephalic. Bi means 2, cephalo means head. So their shape is just like this. They have 2 types of head, like this. Okay, so it appears like they have two types, two number of heads, not two types, they have two number of heads. Why? Because this portion of rib attaches to sternum. It attaches to sternum. Okay, so to sternum, it attaches with the help of a cartilage, and there is only single articulation. Articulation means joint, or the place, number of joint depends upon number of bones. So there are only one portion of sternum and one portion of ribs, and they join like this. So it has only one side, right? Whereas it has two heads or two sides, because the thoracic vertebrae also have two portions for attachment. Like if I say this is a thoracic vertebrae, so it also have two articulation points. The thoracic vertebrae also have two articulation points, and this also have two heads. This also have two heads. Right? So this is how we say the ribs, they are bicephalic. If we talk about the types of ribs, ribs total are 12 pairs. How many pairs? 12 pairs. So we have three types of rib. One are vertebrosternal ribs, also known as true ribs. The first seven pairs of ribs, they are vertebrosternal ribs. Why? Because we say rib cage is formed by thoracic vertebrae, the ribs and the sternum, right? So second type, okay, I will explain you why they are vertebrosternal. Let's just see what are the various types, okay? So you have to keep some suspense. Then we have vertebrochondral. Vertebrochondral ribs are false ribs. What are they? False ribs, and they are 8, 9 and 10 pair. Okay, out of 12, 8th, 9th and 10th pair, they are vertebrochondral or the false rib. Then we have just vertebral ribs, also known as floating ribs. And the floating ribs are 11th and 12th pair. Now, why they are known as true, false and floating? Let's see. So, first of all, how the rib cage is formed. If this is a thoracic vertebra, this is a sternum, and these are ribs, this is how they are formed. On the dorsal side, in the thoracic, these thoracic vertebra, they form the dorsal side of the rib cage, and ribs, they are present laterally, and here on the ventral side is present sternum. This is how rib cage is formed. Now let's see how the sternum looks like. Sternum, this is your sternum. On the upper surface, sternum has this notch or depression. You call it as suprasternum notch. What do we call it as? Suprasternal notch. Suprasternal notch, right? This portion, this one is suprasternal notch. Okay. Now on the sides, these are the notch for your this bone known as collarbone. Okay. So these are the notch for the collarbone. So we are not going to talk about them. Then comes the facets. Facets are also the depression where your ribs got attached. As I've told you, ribs are attached on this side to the thoracic vertebrae and to this side to the sternum. So this is the first facet for the first rib, and then comes the facet for second rib, third, fourth, fifth, sixth and seventh. Second, third, fourth, fifth, sixth and seventh. Right? And this is the last portion. So this is the facet for first rib. Facet for first rib, right? And this is the facet for second, third, fourth, fifth, sixth and seventh rib. So the first pair of ribs are vertebrosternal ribs. They are true ribs. Why? Because dorsally they are attached to thoracic vertebrae and ventrally they are attached to sternum. So that's why they are known as true ribs, right? So if you'll see in this diagram, this is how the ribs are attached. This is how, okay, let me just broaden this up. This is how the ribs are attached. Like this. Okay, like this. So first 7 pair of ribs, they are directly attached to the sternum, and on the back side, every rib is attached to the thoracic vertebrae. That is not a point of difference. The same thing in all the ribs is they are all attached to the thoracic vertebrae dorsally, but the point of difference is in the ventral portion. In ventral portion, where are they attaching? The first 7 pair of ribs, they attach to the first 7 pair of ribs. Where do they attach? They attach directly to the sternum. Okay. And here a cartilage helps. A cartilage helps for the attachment here. which is a costal cartilage. It's a type of hyaline cartilage. It's a type of hyaline cartilage, right? So this is a costal cartilage. It's a type of hyaline cartilage. Okay, now the 8th, 9th, and 10th ribs. Dorsally, they are attached to the thoracic vertebrae, but ventrally to the cartilage of 7th one, and ventrally to the cartilage of 7th one. As you can see, they are not attached to the sternum because they don't have extra facets, right? So these ribs are the false ribs. These are false ribs. So false ribs, guys, dorsally they attach to thoracic vertebrae, but ventrally they attach to cartilage. That's why the contra word is there, because contra word is always used for cartilage, right? So they attach to cartilage of the 7th rib. Okay, what about vertebral? They are not at all attached to the sternum at all. They are just floating in the air, and they are only attached dorsally to the thoracic vertebrae. So this is a most common question that is asked in the exam about the different types of ribs, right? So you can see here the last pair, 11th and 12th. Let me label all the numbers for your better understanding. This is the first rib, 2nd, 3rd, 4th, 5th, 6th, 7th, 8th, 9th, 10th, 11th and 12th. These two are floating ribs. These three are false ribs, and above all, they are true ribs from 1st to 7th. Okay, so this is how you have different types of rib forming the rib cage. All right, guys, so that's about the these bones, ribs and sternum. We are going to talk about the vertebral column now. Vertebral column, commonly known as backbone. Commonly known as backbone. What it is made up of, and first of all, what is its function? It provides support to your body. It provides flexibility to your body. I can move down, up, and my erect posture is just because of this vertebral column, right? So it provides support to body. Inside the vertebral column, if you'll see the vertebrae, let me show you. Imagine this is one vertebra. Inside the vertebra is present this small cavity. Okay, so when all these vertebrae, they stack upon each other, when they stack upon each other, for example, all these vertebrae, they are stacking upon each other like that, and all have an empty space in between them. They will form this canal, the empty space between the empty space inside the vertebral column. You call it as neural canal. So we say there is an empty space inside this vertebral column, and that contains your spinal cord. That contains your spinal cord, right? So empty space inside vertebra, it forms what? It forms neural canal, and this neural canal, it contains spinal cord. So I can say that my backbone is protecting my spinal cord, right? So it contains what? It contains spinal cord. So next function of this is it protects spinal cord. Another point to note here is that thoracic vertebrae are also attaching my ribs. So they also helps in attachment of ribs. Attachment of ribs. Not the entire vertebral column, but the thoracic vertebrae of it. What are thoracic vertebrae? I'll tell you that in detail too. Then it also supports head. So entire head is supported, right? Second, it also helps in the attachment of muscles. All your back muscles are also attached to your backbone. Now, what your backbone or vertebral column it is made up of? So, vertebral column is made up of two things. What are these two things? One are the vertebrae, which are small irregular bones, total 26, and in between present is vertebral or intervertebral disc. What is present in between them? Intervertebral disc. Okay, let me show you. For example, this is one vertebra, this is second vertebra. In between them is present this cushion-like substance known as intervertebral disc, and this intervertebral disc is made up of cartilage. It provides support. You must have heard of slip disc. A person falls off and got slip disc. In slip disc, this cushion, it gets displaced from its original location, and nerves got pressed, and it's really very painful. Fine. So these vertebrae, they are of various kind: cervical, thoracic, lumbar, and in the pelvic region we have sacrum and coccyx. Okay, we have 7 cervical, 12 thoracic, 5 lumbar, 1 sacrum, and 1 coccyx. These are the various types of, or numbers of vertebra or small bones that we have, right? Sacrum and coccyx in the embryonic stages, sacrum were total 5 in number and coccyx was 4 in number in the embryonic stages. But later on in the adult, these get fused to form a single bone. Coccyx is your tailbone. Coccyx is your tailbone. Fine. So these are the various types of vertebrae. So the one which are present in the thorax, you can see, they are the one which attaches to ribs. Okay. All right. Moving further to the first cervical vertebra. First cervical vertebra, cervicals are in the neck. That is known as atlas. And the second is C2, as it is written here also, that is axis. Atlas was in the Greek mythology. That was a person or god, I don't know exactly. He was the one who, it is believed that he has entire earth on his shoulders, and he has hold the entire earth on his shoulders, right? So whenever he changes the hand when he got tired, then it leads to the earthquakes and all. This was what people used to believe earlier, right? So just like that, Atlas is also bearing the weight of entire head. So that's why the first cervical vertebra was named as Atlas. So Atlas is a one that forms joints. One joint is formed between this portion of axis and atlas here. You call this joint as atlantoaxial joint. So there are like C1 vertebra, C2 vertebra, and there is head. This is how these are located. Okay. So in the head, we have occipital condyles. What do we have? Occipital condyle. These occipital condyles, they form joint with the atlas, and you call this joint as atlanto-occipital joint. Okay. Now, second joint is formed between C1 and C2, that is atlas and axis. You call it as atlantoaxial joint. Atlantoaxial joint is a joint that helps you to say no, and this one helps you to say yes. For example, these are two occipital condyle, right? And here is present atlas, and this is how it has fixed. Or we can say on these processes, your occipital condyles got fixed. You can see this, right? On these processes, your occipital condyles got fixed. So whenever I am saying this yes, like this, at this point, these two bones have been articulated. That means they have been fixed, they have formed the joint, and they help me to say yes. When I'm saying no, at this point, if this is axis with its den and this is atlas, they are moving in this direction and help me to say no. Okay, so now you know how you say no and how you say yes. Okay, all right. So that's about the vertebral column, guys. Let's move further and talk about the appendicular skeleton. In appendicular, we have around 126 bones. Out of 126 bones, 120 are the part of the limbs, and 6 bones, they are the part of girdles. Okay. So what are girdles? Girdles are those part of your skeletal system to which your appendicular skeletal, or you can say the limbs, attaches. You know, limbs can be hanging like that. So they need something to get attached here, or in case of the lower limbs, the hip is the portion where your lower limbs get attached. Okay. So for that attachment, we needed some girdles. Girdles of the upper body, they are pectoral girdles. The lowers of the lower body are the pelvic girdle. Okay. Pelvic girdle is just made up of 2 bones, and pectoral is made up of total 4 bones. Pectoral have 2 types of bones. Scapulae, they are 2 in number, and clavicle, they are also 2 in number. So clavicle are your collar bones. These one, they are 2 in number. Yes, you can see that there are two in number. You can't see mine because I'm little fatty, right? So clavicle, they are also known as collarbones. Collarbone or beauty bone. Now, whereas scapulae, these are your shoulder blades present right here. These are your shoulder blades. Pelvic is formed by hip bones. How many hips do you have? 2. Hip bones, they are also known as coxal bone. And another name of hip bone is innominate bone. These are the 3 names for one single, right, bone. All right. Now, what about limbs? We have 2 types of limbs. Forelimbs, which are of the upper portion, and hind limbs, which are of the lower body. Total 30 bones are present in one limb, and we have 2 hind limb and we have 2 forelimbs. So total, this is 60, this is 60, 60 plus 60 is 120. So I'm good, so good in maths. This is the only maths I know, I tell you. All right. So that's about your appendicular skeletons introduction. So first, we'll talk about the pectoral girdle. In pectoral girdle, guys, okay, before pectoral girdle, I think we should talk about limbs first. Then the things will be more clearer. Okay. So first, limbs, as ma'am says. So forelimbs, what it is made up of? Let me tell you. What are the various bones of forelimb? First we have is humerus. How many? 1. This bone is humerus. This bone is humerus. Then come to this portion. This one which is present towards little finger is ulna. The one which is present towards thumb is radius. Radius ulna. Then in the wrist we have carpals. Then here we have metacarpals. And in the fingers we have phalanges. What do we have? Phalanges. So we have 1 humerus, 1 radius, 1 ulna, total 8 carpals which are present in the wrist, metacarpals which are present in the palms. How many metacarpals? 5. Why? Because they are all emerging towards your fingers. You have 5 fingers. And phalanges, 14. F for phalanges, F for fingers, and F for 14. This is how we speak. I'm not talking about the writing spellings, okay? Though it is written with P, but you say with F, okay? This is how its pronunciation is. All right, guys. So here you can see in the human upper arm contains humerus. Humerus is forming joint with both radius and ulna, because here you can see this is the first finger, that is thumb. So this bone is your radius. This bone is your ulna. Okay, this is radius, this is ulna. Now in the wrist you have these small bones known as carpals. This is how the carpals are arranged. Total 8, 4 on this upper row, this which is a proximal row, and 4 on the distal row. What is distal? That is away. Proximal, which is towards. We have done this in the PCT and DCT also. Okay. So here these are the various names of the carpals. If you can learn it very well, if you cannot, don't pressurize yourself, right? So here scaphoid, lunate, trichotrum, pisiform, trapezium, trapezoid, capitate. If you cannot see the name, I can write it for you. Capitate and hamate. So how can you learn this if you want to? She looks too pretty. She looks too pretty. Tri, that means trapezium. 2, trapezoid. Capitate, catch. And her is hamate. You can learn like this. She looks too pretty, tight to catch her. All right. That's about forelimb. Let's talk about hind limb. In hind limb, what are the bones we have? First of all, the number is same. 30 in each. Okay. 30 here, 30 down. We have one thigh bone, which is femur. As you can see here, this is femur. Then downside, what do we have? We have knee. After thigh, what do you have? Start looking at your body also. Then we have knee. In the knee, we have kneecap, also known as patella. 1. So this was not present in the forelimb. Then bones are total 30. Where 1 is going to adjust. Let's see. Then just there we have radius and ulna, just like that. Here we also have tibia and fibula. We have tibia and fibula. 1, 1. As you can see, fibula is a very small thin bone as comparison to tibia, right? T for toe toe is the thumb of your feet, and T for tibia. So it is present towards toe. As you can see, tibia is forming joint with the femur, and also femur is forming joint with this kneecap patella. Fibula does not form any joint with the femur. Fibula do not form any joint with the femur. Okay. Then after that we have tarsals. There we have carpals, which were 8 in number. We have tarsals, 7 in number. Then we have metatarsals, guys. Metatarsals, which are 5 in number. And then we have phalanges. Phalanges are total, again, 14 in number. How are they arranged basically? How phalanges are arranged in both hand and feet? 2, 3, 3, 3, 3. 1, 2, 3, 4, 5 fingers we have. This is for thumb. And these are for other fingers. Same for the forelimb. Okay. Now, as you can see, femur have a head here. Just like that, humerus also have a head here. This portion of humerus is head. Alright. So, these head are attached to the girdles. These head, they are attached to girdles. They are attached to girdles. Because first we have studied about the forelimb, so we'll study the pectoral girdle first. Pectoral girdle have two bones. One is scapula and another are clavicles. Okay guys, now this, which scapula, it's a triangular shaped bone. It's a triangular shaped bone. Just like this. Okay, this is how it looks like. And here it has small, ridge-like structure. This is very, what you say in Hindi, ubri hui. It's a ridge-like structure and prominent structure, we can say in English. And it forms spine. If you just touch your scapula, is something you can feel it, which is very spine-like. So that's the spine portion of your scapula, which then gets broader and form this head-like structure. You call it as acromion. What do you call it as? Acromion. So this portion is acromion. Acromion, right? And this is spine. This is glenoid cavity. On this glenoid cavity, the head of humerus fits. I told you the head fits somewhere in the pectoral girdle. So where does it fit? It fits in this glenoid cavity. So head of humerus fits here. Okay. Now, two acromion clavicle joints, right? So clavicle on one hand, it joins to sternum here. On this surface, it attaches to acromion. So to acromion, clavicle joins here. All right. Moving further to the pelvic girdle. Pelvic girdle is formed of two hip bones. It is formed of two hip bones. Okay. So if you'll see to the dorsal side, the sacrum is present. To the dorsal side, this is how sacrum is present. Where it is present? To the dorsal side. Sacrum is attached on the upper side. What is sacrum, guys? You know what is sacrum? What is sacrum? The bone of vertebral column. To the sacrum, here is attached the lumbar vertebrae. What is attached here? Lumbar vertebrae, right? On this portion is attached hip bones. So hip bones are not the flat bones, rather they are curvy bones like that. They are curvy bones like that. Okay, let me just draw this for you. They're curvy bones like that. Okay, they are not flat bones, rather they are little scooped. They are like this. They are like this. Okay, so if this is, imagine my face is sacrum. So to the dorsal side, it is attached to the sacrum like this, and to the front, it is coming like this, and between these two bones is present a white fibrous cartilage. What is present? A white fibrous cartilage. So white fibrous cartilage, first it is present in the intervertebral disc, and second it is present between the two hip bones. So here is present a white fibrous cartilage, and this portion also have a name, like intervertebral disc have a name, just like that. Its name is pubic symphysis. Pubic symphysis. So pubic symphysis is made up of white fibrous cartilage. So it's also a type of a joint. As I've told you, the cartilages are usually present in the joints. Okay. All right. Next. So what a hip bone is made up of? You can see this diagram. Okay. So one hip bone is formed by the fusion of three bones. One hip bone is formed by the fusion of three bones. Now what are these three bones? Ileum. So this is not the ileum of digestive system. The ileum of digestive system is ileum. Ileum, then ischium, and then pubis. So let's see this one. So this yellow one is ileum, this is sacrum, and here is your pubic symphysis, guys, right? This is coccyx, this is lumbar vertebra, L5. Total how many lumbar are there? 5 vertebrae, right? So this is L4, this is L3. So this is ileum, this is pubis, this is ischium. Okay. Now where all these three bones are fused, you can see here ileum is also having a connection, pubis and ischium. This is the point known as acetabulum. Where all the three bones fuse, there is a portion known as acetabulum. It's a depression-like portion, just like glenoid cavity. And to this, the head of humerus attaches. And to this, the head of humerus attaches, right? So here, the fusion of three bones. There is a portion known as acetabulum. And to acetabulum, head of femur fits here. Alright, so you can see this is the head of femur. This is head of the femur, and this portion depression is acetabulum. Okay, alright. Moving further to the joints, the most important topic. Alright guys, so let's talk about joints now. What is a joint basically? It's the articulation between two bones. Articulation means the closeness, okay, where both have been integrated like this. So joints are the articulation between two bones. There are three types of joints. How many types? Three types. One is the fibrous, also known as fixed joint. Second is cartilaginous. And third, your synovial. Okay, write this down in the copy. In meanwhile, I drink some water. All right. So, they have been classified on the basis of how much movement can be allowed around these joints, right? For example, these joints, they're very fixed. So they are not at all mobile. That means they are fixed. They are immobile. Or we can say no movement. Right? Whereas cartilaginous, they allow a little movement around the bones. Okay? So they can allow slight movement. And this one, they are highly movable joints. For example, right now I'm talking to you, my jaw is moving, that's a synovial joint. My arms, my elbows are moving like this, that's also a synovial joint, right? So this one contains white fibrous cartilage. As you all know, the bones, when two bones join, they form a joint. So for or in the union of that bones, there need to have a cartilage. So here we have white fibrous cartilage. The example is sutures of skull. All right, let me show you. So this is a diagram. Okay, so here you can see these lines where two bones are joining. These are sutures. These are what? Sutures. Okay. Next, cartilaginous can have either white fibrous cartilage. And right now we have done two examples. One is intervertebral disc, that means between two vertebra. We have cartilaginous joint with white fibrous cartilage and between two hips, that is pubic symphysis. So that's a cartilaginous joint with white fibrous cartilage. Also, it might have hyaline cartilage, and that we have done right now. Tell me the example if you can, if it strikes in your mind. Where have we done hyaline cartilage till now in this chapter? Between, yes, ribs and sternum, costal cartilage. Okay, then the synovial joint. How synovial joint is formed? It's formed between two bones. Imagine these are two long bones, right? The ends of long bones, they are covered by articular cartilage. Why nature is doing so? Why are long bones, or the bones specially which are involved with synovial joint, why do they have the coating of articular cartilage, which is also a type of hyaline cartilage? The reason is, imagine, your calcium is present in the blood. There can be a possibility that if this cartilage was not present, the entire calcium will be pasted on the bone and the distance between two bones will be reduced, and we don't want to do that. Imagine the distance between these two bones joint reduces. The movement will be very difficult. Imagine you have two sticks. Okay, I don't have here. Okay, let me show you something. Okay, just imagine this is one bone, this is another bone. If I just join them completely like this with a tape, will they be able to move? No. If I kept a little distance like this and I want them to move, they can easily move. This is how synovial joints are. They have a little space in between them. They have no space in between them. The bones, they do not have any space in between them. Here, the bones have little space in between them. Here, the bones have large space in between them. That's why they are highly movable joints. So now, two bones, they are joined to each other via ligament. So both bones will be attached to each other via ligaments. Okay, so these are what ligaments are. Now, on this portion is present a membrane known as synovial membrane. What this membrane is? Synovial membrane, from where the joint got its name. This synovial membrane, it secretes a fluid known as synovial fluid. What fluid? Synovial fluid. And this synovial fluid, it is rich in hyaluronic acid and it reduces friction so that the bones can easily move. They can easily move. This is what your synovial joint is, or how it looks like. Now let's talk about the types of synovial joints. Let's talk about the types of synovial joints. Types of synovial joints. All right, guys. First type of a synovial joint we have is ball and socket. So what happened in the ball and socket? We have this socket-like structure, and then we have a ball, and this ball can move in all the directions. Like this is a socket and this is a ball, and it can move in all the directions like this. So this type of a socket we have seen in the pectoral girdles glenoid cavity, and we have also seen in the hip bones acetabulum. So this type of a joint is present in hip joint and shoulder joint. Okay, how to remember this? Ball. How do you in cricket, how do you throw the ball? Like this. So for throwing the ball, this joint is working. So this is ball and socket. Why it is how this joint has been formed? Between the glenoid cavity of scapula and head of humerus. Same example is also seen in the leg. In the leg, we have the acetabulum and the head. Okay. Now, second type of a joint is hinge joint. In hinge joint, it's just like the door and the wall. The space between the door and the wall is the hinge. Okay, that's the hinge. So in the hinge joint, the movement can be allowed in only this direction, like this. Okay, so where do we have? Like this direction, elbow joint. So the example of the hinge is elbow joint. Such type of a joint is also seen in your knee. That's a knee joint. All right. Next example that we have is your saddle joint. Saddle joint. Saddle joint is a joint which is a poorly developed ball and socket. Poorly developed ball and socket. This is a joint which wanted to be a ball and socket, right? Just like a child who wants to be doctor. But if the child does not be a doctor, the child will go sad. Okay, so this is also a sad joint. Sad because it wanted to be a ball and socket. Similarly, you have this hand. Okay, in this hand, you have here carpals. Okay, and then we have metacarpal. So the joint here between carpal and metacarpal only that are going towards the thumb is your saddle joint. All the fingers are together, but the thumb is alone. So it also becomes sad. So saddle joint is present between carpal and metacarpal of thumb. Right, guys? Then we have fourth type of a joint, which is a gliding joint. In gliding joint, two bones are generally flat like this. Two bones, they are generally flat and they glide upon each other like that. They glide upon each other like that. So it's present in the carpals. You all know the carpals. Imagine if this is one carpal, this is another carpal. Both are flat bones and they can glide upon each other. Same like that, we have tarsals. We have tarsals. Okay, tarsals also, they glide upon each other like that. And then we have ellipsoidal joint. Ellipsoidal or condyloid. In this type of a joint, the movement is either in this direction or in this direction. Okay, let me show you. Like this or like this. Okay, like this or like this. So such kind of a joint is your ellipsoidal, or you also call it as condyloid or angular because it's in a certain angles. So where do you find this one? You find this in the Atlanto-occipital. Atlanto-occipital. Now the last type of the synovial joint. That is, okay, I'm writing it here, pivot joint. In pivot joint, guys, one bone is fixed like that, okay, and other one is moving. Imagine this is the second one. So this one is moving in this direction. So where do you find this type of a joint? With this type of a direction, guys, where do you find it? Atlanto-axial. And also between radius and ulna. You heard it right. There is also a joint between radius and ulna. So that's a type of a pivot joint. So these are the various types of joints. Which joint? Synovial. Okay. So that's all about the skeletal system. It's time to learn about disorders, guys. Now, what are the disorders associated, first of all, with muscles? With muscles, we have three types of disorders or three disorders. One is muscular dystrophy. Second is myasthenia gravis. And last is tetany. Let's get started with this. All right. So muscular dystrophy is a genetic disorder. It's a genetic disorder. That means the genes are not fine. It's a genetic disorder which will lead to muscle degeneration. Which degeneration? Skeletal muscle degeneration. Skeletal muscle degeneration. The muscles will die. Why? There is a gene of dystrophin protein. If you remember, we have done a dystrophin protein that holds the myofibrils and attach it to the sarcolemma. If that goes wrong, the myofibril will be, pushed here and there and the positions will be changed and muscle will not be able to contract properly. So that's the muscular dystrophy, leads to muscle degeneration and even paralysis. Now, in the myasthenia gravis, it is autoimmune disorder. What is autoimmune disorder? Autoimmune is a disorder where your own body cells are attacked by your own immunity. Now, which cells? This was a motor neuron and this was your muscles, right? In muscles, we have these sodium ion channels, right? To these sodium ion channels were attaching the ACH. Now, ACH cannot attach to it. Why? Because they are attacked by antibodies. They are attacked by antibodies. So now they are attacked by antibodies, ACH cannot bind, and then there will be no muscle depolarization or excitation, guys. See, if you do not remember, I am there to make it clear for you guys. Okay, so this one runs really very fast. All right. Where are you, muscle? Okay, so here what was happening, ACH was binding here, but what if antibodies attaches here? Sodium ion will never enter because the channel will never open, and no muscle excitation, no calcium ion, no muscle contraction. So this is what it happens in your myasthenia gravis, right? So in the myasthenia gravis, it is an autoimmune disorder. The antibodies are formed against ACH receptors, acetylcholine receptors on muscles, and which will lead to muscle degeneration, muscle paralysis, and then muscle degeneration. The muscle will die. Okay, such kind of people, even not able to engulf the food properly because skeletal muscles are there on the upper 1/3 of esophagus and in the pharynx. Okay, tetany occurs whenever you have low calcium ion in the body fluids. So whenever you have low calcium ions in the body fluids, that will lead to rapid spasm of muscle. What are spasm? Wild contractions of muscle, right? This is what tetany is. So this was tetany and muscle disorders. Let's talk about the disorders of skeletal system. First of all, we have arthritis. What is arthritis, guys? Arthritis is inflammation in joints. Inflammation in the joints. Okay. Now, this inflammation, you also call in Hindi as sujan. Inflammation have occurred due to which it leads to the restricted movement of joints. The joints are not able to move it, right? For example, you must have seen that in old people, they're not able to, walk properly because their knee joint is affected. Or you have seen their hip joint is affected, especially in females, right? So what happened in arthritis? Arthritis is of 3 types. One is rheumatoid. Arthritis. Second is gout. And third is osteoarthritis. So in osteoarthritis, this one occurs due to aging, and you can see the cause is aging, and you can see usually hip and knee joints are affected. With age, this synovial membrane and articular cartilage, they degenerates, and that's how the joint degenerate and the person is not able to move properly. So osteoarthritis occurs due to aging. The gout occurs due to excessive accumulation of uric acid. If there is more uric acid in your blood, that can due to you eat a lot of pulses, and so the uric acid can be increased. That can also damage your joint and cause inflammation. Or if your own body immunity is attacking your joints, and that's your rheumatoid arthritis, which is autoimmune. So arthritis is same. The result will be same, but the cause is different. Like in rheumatoid, usually your hands are affected, right? In rheumatoid, it's autoimmune. Whereas in gout, uric acid gets accumulated, and in osteo, it occurs due to aging. One such another disorder is osteoporosis. Osteo means bones, poro means pores, and cis means synthesis. So there is pores in the bones. Why? Due to low estrogen. So when a female after around 40 age, or we can say between 45 to 50 of age, the female's menstrual cycle, it stops. When the menstrual cycle of female stop, her estrogen level goes down. And estrogen have a very important role in females, that it helps in the deposition of calcium on your bones. For example, I'm a female, I have a lot of estrogen, okay, then that is helping me for my bone growth, or putting the calcium on the bones, which is calcium deposition. But if when I'll hit the menopause around in 50s age, then my estrogen will go low, Now, who will deposit calcium? No one. So now, in fact, the calcium will be, the bones will get demineralized and calcium will be taken out from the bones. As a result, the pores will be formed in the bones, known as osteoporosis. So after menopause, female have the more chances of fracture because her bones got thin, and also there are more chances of arthritis. So usually females get, I tell you, osteoarthritis. Okay, all right. So that's all about the chapter locomotion and movement. So before that, I think let's read the NCRT of these disorders, because this is really very important. Questions are asked from this topic every year. Okay, disorders of every chapter is important. Myasthenia gravis, autoimmune disorder affecting neuromuscular junction leading to fatigue, weakening and paralysis of skeletal muscle. This is what we have done. The antibodies are formed against ACH receptor, and that's a part of neuromuscular junction. Muscular dystrophy, progressive degeneration of skeletal muscle, mostly because it's a genetic disorder. What genes are affected? The genes of dystrophin protein. Tetany, rapid spasm in muscle due to low calcium ion in body fluid. Arthritis, inflammation of joint. Osteoporosis, age-related disorder, again age-related, after 50, around 45 to 50, characterized by decreased bone mass decreases, and increased chances of fractures. Decreased level of estrogen is a common cause. Gout, inflammation of joint, it is also an arthritis due to accumulation of uric acid crystals, right? So that's about the locomotion and movement chapter, guys. So I hope you will study this well. And let me know if you have any, suggestions if you want to give me. And if not, that's okay. Or you can send me hearts and lots of love. So that's it about locomotion and movement. Next will be a very, very important chapter, neural control and coordination. And we will hit that one, and we'll try to, do it. The entire NCRT part will be covered there. I'll try that entire NCRT part will be covered. And all I want is that a promise from you guys that you will also read entire NCRT of other chapters too. See, in locomotion, the skeletal portion is very easy from NCRT. That's why I didn't make you read here. I know my students can read it themselves. So that's all about locomotion. Lots of love, guys. Lots of love. And I hope I'll get well very soon. And my voice will be back just like that. I think now it's improving a little. But anyways, so study hard. Work for your goals. Work for what you aim for. And do not be afraid. You have everything. You have your teachers with you, you have your books with you, you have your brain with you. The only thing that we need right here is your determination. That's it. Doesn't matter from where you come from, doesn't matter you have money or not, your parents have money or not, doesn't matter. Nothing matters if your hard work comes forward and your determination along with that. That's the only thing that will keep you going, and that's the only thing that will help you to succeed. Mind it and mark my words, hard work and smartness and determination together, they can do anything. They can help you to become a doctor or IAS or whatsoever it is. So bye bye, take care. And the season is changing, so take care of yourself. It's a rainy season somewhere, so take care of yourself and take care of your health. Eat nicely and study hard.